All right. Hi, guys. So today we're doing hypersensitivities, and this is also going to include transfusion reactions, transplants, and we're going to go into blood typing a little bit. You definitely should know blood typing. They love, love, love to ask about blood typing on immuno, like for as immuno questions. Okay, so let's jump in. So we're going to talk today about type 1 through type 4 hypersensitivity reactions and how you know if it's type 1, type 2, type 3, type 4, etc. And a little bit into like how the PPD test works because that's kind of like what they like to ask about. If you see HSR, I'm referring to hypersensitivity reaction. Jumping into transplant transfusion reactions and then blood typing and hemolytic reactions and then ending with some questions. Okay, so disclaimer. So we haven't formally talked about allergies yet. So a quick disclaimer is that no one actually knows what causes allergies or like why do some people have allergies and other people don't have allergies. So for an example, why does cat dander stress out my immune system, but I have a twin brother, right, has no issues with cat dander. So like they don't really know what causes it. Some theories, they think it has something to do with exposure as a child. So for example, they think if you're a C-section baby, you're not being exposed to the vaginal micro like the flora, like the microbes that are within the vaginal canal. So they think maybe that has something to do with it. Maybe breastfed babies versus non-breastfed babies, whatever. This is known as the hygiene hypothesis. We're going to talk about that on the next slide. Or they think there's specific HLA types that are just predisposed. So remember, I kind of showed you that first aid page and was like, you should know like which of these HLA types are associated with certain diseases. That's just like correlational, but they think that that could be what's causing it. And remember, HLA are the genes that code for your MHCs. So if it's an HLA with a single letter, so like HLA A, B, or C, that's going to be for your MHC class ones. And then if it's two letters, so HLA, DR, DQ, D whatever, those are your MHC class twos. But again, theories are theories, so nothing's proven, but it's kind of cool to think about like what causes it. But just keep this in mind as we like move forward because we're gonna go into like a little bit with like allergies and stuff. So don't memorize this. This is just kind of showing you what I was kind of talking about. So these are the two leading theories as to like what causes allergies and like even autoimmunity in general, but specifically allergies. So it's known as the hygiene hypothesis. And all of this is basically saying that it's like if you're super clean as a kid, like really, really clean, like you're never being exposed to anything, then your immune system's gonna just start freaking. It's gonna, because the immune system thinks that there should be pathogens around. So if you're really clean and you're never being exposed to pathogens, then maybe it's freaking out. It's like we need something to do. So that's where allergies might be coming from. Goes back to like again C-section versus the vaginal birth, babies, things like that. So that's the hygiene hypothesis. After I learned about this, I know I'm gonna be throwing my child in dirt when they grow up. I'm just gonna be like exposing them to all the microbes so they don't get allergies. But other instances are genetic susceptibility. So again, here's what we were talking about, like HLA, DR, DQ, DP. So this is kind of going to be your MHC class 2 allele, like protein. So this is why it's like involved with antigen presentation. Some of these other ones, so the immunoregulatory cytokines, my people that watch the T cell lecture, you better know like IL-10, TGF beta, these are your T regulatory cytokines. This is what they're secreting that dampens the immune system. Again, don't memorize this. But I just kind of wanted to point out this is what they're thinking is like causing susceptibility to developing allergies later in life. Okay. So basically, a hypersensitivity, when you hear this term, think in some way, shape, or form this is an allergic reaction. It's the body, it's like an immune reaction, like the immune system is reacting to something. So types 1 through 3, these are known as antibody mediated. So type 1, type 2, and type 3 are all mediated by antibodies. And what that means is antibodies are what's causing the damage. In some way, shape, or form, antibodies are causing damage. Type 4, this is T cell mediated. And I'm going to show you like when we do questions at the end how you can kind of identify like if it's going to be a type 4. There's always going to be like little clues in the question. But this is like enough to diagnose it as a type 4. If a T cell is involved, it's enough, okay? Whereas 1 through 3, the antibodies are what's doing the damage. So type 1, this is specifically, and we're going to go way into more detail, but type 1, this is specifically mediated by IgE. We're going to talk about that. That causes mast cells to degranulate. Type 2 and type 3 is mediated by IgG. And this is where you kind of get complement mediation, you get like phagocytic effector mechanisms. And what I mean by that is like IgG. Remember, IgG can kind of coat the surface of whatever it binds with its FAB portion. And then, NK cells can come in, do ADCC. So ADCC can be involved here. Macrophages have Fc gamma receptors, so they can kind of bind it up because it's acting as an opsonizer. So things like that. Type 2, though, so the difference between type 2 and 3, because it's the same antibody that's involved, type 2, and again, we're going to go into this, think it's like the antibody is binding to like a antigen that's fixed. So the antigen is going to be like fixed to a surface, and then the antibody binds to it. Versus type 3, which you have antibody-antigen complexes circulating around, and then they deposit places. And again, one of the ways that we talked about complement getting activated, the classical pathway, is with antigen-antibody complexes. So it's all sorts of like, you get a lot of issues with the type 3s. But that's the main difference between, otherwise they're pretty much the same. Type 2 is just like the antibody's being directed against a non-moving antigen, versus type 3, like now there are these complexes and they're kind of circulating around and then they deposit, and that's what's causing the issue. So then type 4, this is your T cell mediated, so we're going to go into this, but remember Th1 T cells are what activate your macrophages, so you get this very big inflammatory response. Th2 mediated, this is kind of your activation of eosinophils. Again, we're going to go into this. And then your cytotoxic T cell mediated. This is like another abbreviation for cytotoxic T cell. I think it's like cytotoxic lymphocyte. So if you see CTL, that's like another name for it. So I just wanted to make it 3 letters. Okay. But cytotoxic T cell mediated, this is the damage is directly caused by the cytotoxic T cells. So it's like they can either indirectly cause damage from this inflammatory response, indirectly cause damage from activating eosinophils, and then the eosinophils release their mediators, or the cytotoxic T cells directly causing the issues. Okay, so this is kind of like an overview of what we're going to go into. This is from first aid. I really liked this. This is like a good way to memorize it when you're first learning them. So think of it as hypersensitivities are A, B, C, D. So like type 1, type 2, type 3, and type 4. So the A is anaphylactic and atopic. Again, we're going to talk about all of these. Type 2, antibody mediated. I mean, the first 3 are antibody mediated, but that's fine. Type 3, think complexes. And then type 4 is delayed. So another name for type 4 hypersensitivity is delayed hypersensitivity. And again, it's delayed because you're getting T cells involved. But this is like a good way, at least at first, to think of like hypersensitivities. ABCD, anaphylactic, antibody, complexes, delayed. And then here's a little bit more detail. I really like this chart. It has like everything on here. And of course, we're going to go into more detail. But here are the 4 types along the top. So type 1, again, remember it's IgE mediated. It's always going to be IgE. Don't worry so much about like the type of antigen. This is more important for like, type 2 versus type 3. But we have basically mast cell being degranulated. That's what's causing the symptoms. So think anaphylaxis, think atopic allergies, think things like that. Type 2, there's different types. We'll talk about them, but IgG is the guy that's doing it. And remember, it's always going to be against something that's like fixed. So cell surface receptor is one example of it, but it can also just be like a fixed antigen. And that can be a type 2 versus type 3, which is also IgG. But now it's like we have those complexes. So we have these circulating immune complexes. So of course we get complement activated like immediately. And then type 4, like I said, there's the 3 types. Th1 cells involved, think macrophages, so very inflammatory. Th2 cells are involved, think eosinophils. So this is like what's going to happen with like the eosinophil degranulation. And then your cytotoxic T cells, this is just cytotoxicity. And we'll go into like when you see this. But when you think like seasonal allergies, like what people are probably experiencing right now, that's your type 1. This is like your classic allergies, is type 1 hypersensitivity. Okay, so first we're going to talk about type 1 hypersensitivities. I tried to put like pictures to help you like remember all the different hypersensitivity responses. But type 1, this is your immediate type. So it's immediate type, meaning that it's going to act immediately, which again, if you go outside and you breathe in and you're allergic to pollen, you sneeze immediately. So this is like examples of just when you think type 1 hypersensitivity, this is kind of what you should be thinking of. You should be thinking of mast cells. You should be thinking of things that make people sneeze. You should be thinking of like this wheel and flare reaction, which we're going to talk about. But this should all be associated with type 1. So a notable difference between B cells and mast cells is that mast cells can bind IgE of many different specificities. Yeah, so something that I wanted to kind of point out is that see B cells, how let's say this B cell has, it's going to have the same B cell receptor on its surface. It's not like it's going to have one anti-herring worm B cell receptor and then another anti-blood fluke worm and then anti-hook worm. Like B cells don't do that. They have one receptor type on their surface. That's it. That's the point of B cells, right? Like they want to recognize one specific antigen, and that way they can undergo somatic hypermutation, right? They can undergo this, make a really, really tight fit, and then have memory and have plasma cells. Mast cells can do this. So mast cells can have IgE antibodies specific to many different things. So for example, they can recognize anti-pollen, they can recognize anti-cat dander, anti-peanut, anti-bee venom. So they can have different IgE specificities on its surface. Okay, so this is why someone that has one allergy usually has multiple allergies. So like me, I'm allergic to cats, I'm allergic to pollen, I'm allergic to tons of stuff. So my mast cells have just tons of IgE antibody on their surface for all of these different things that I'm allergic to. So we never really talked about like how mast cells release the histamine, but it's going to be pretty much the same thing as when you were to activate like a B cell. So you need cross-linking. So remember when I said when you have B cells, you kind of need to, if we want to activate the B cell, we need to cross-link the B cell receptors in some way, shape, or form. It's the same thing here. You need to cross-link two IgE antibodies in order to activate the mast cell. Okay, so when we cross-link two IgEs, this is going to go through this whole process, a signal transduction process, blah, blah, blah. And then at the end of the day, you're going to get release of histamine. So these granules contain histamine, so this is called degranulation. And then histamine is what causes the symptoms of allergies. So remember in the innate immune thing when we talked about the inflammatory response and the 5 cardinal signs of inflammation, it's because of histamine. It's causing that dilation of the blood vessels. It's causing the leakiness of the blood vessels because the endothelial cells kind of like move apart a little bit, causes red, swelling, all that stuff. That's a lot of the times it's due to histamine. There's other cytokines involved, obviously, but a lot of the times it's because of histamine. It's causing this, thinks that something, it's like we need to get rid of whatever allergen is here. So type 1 hypersensitivities, 2 examples are allergies and asthma. So this is IgE mediated, another name, immediate type 1 allergy. So I kind of drew over here, again, a mast cell with different types of IgEs in the, basically like sticking on the surface of the cell. Again, mast cells have, this is not as accurate, this is a lot better because it's showing like the Fc epsilon receptor. So mast cells have Fc epsilon receptors on their surface, so they're going to recognize the Fc portion of the IgE antibody. That's where epsilon comes from. So it has these Fc epsilon receptors on its surface that recognize this. And any of the IgE antibodies like that mediate allergies can stick to this surface. So again, anti-cat, anti-pollen, anti-peanut, whatever. This can be, the whole cell can be made up of different types of IgEs. But it doesn't just happen like this. It doesn't just happen where the first time you ever eat a peanut in your entire life, you're going to immediately be you're going to immediately have this anaphylactic reaction. So there's 2 parts that and this is really important. There's 2 parts to type 1 hypersensitivities. You need sensitization. This is absolutely required. So this means the first time you ever see a particular antigen. So if you eat a peanut for the first time, that's your first exposure, you will not have an immune reaction. So some people that are like, oh, I can't be allergic to bees. I got stung by a bee one time and then nothing happened. That was sensitization. That was your first time. You have no idea if you're actually allergic until you see it that second time. Okay? So and think of this as like kind of like you have to let the body make IgE like against that particular antigen. So that's why no immediate reaction is occurring. It's kind of like you're just kind of making the IgE. So again, body treats the allergen as if it's a foreign peptide. So it's great. It's like, great. Like, this is what you want if it was an actual foreign peptide and not like a peanut. But this is what happens. You get memory cells, you get T cells activated, you get IgE specific for the antigen, the allergen, and then it's going to go and stick into the mast cell. So that happens the first time. So again, you have no reaction after you have something for the first time. However, the second time you see it, this happens immediately. So this is your immediate type. So the second time you see that allergen, if you became sensitized to it, you're going to get an immediate immune response. And immediate is like within 30 minutes. That's even like not even that long. I'll give you an example. Like I get allergy shots because again, I'm allergic to literally everything. And they make me sit in the office after I get my shot for 30 minutes afterwards. And it's to make sure I'm not having like this immediate type 1 hypersensitivity. And then after 30 minutes, they're like, cool, you can leave. So it's kind of like within that 30-minute window, you're going to see a reaction. Here's kind of like a figure like showing like what's going on. So this is a lot more info than you need, but I just thought it was cool to kind of see it all come together. So most allergens have what like enzyme capabilities. So a lot of the times you can just think of allergens as being like proteins. So they are. So here's an allergen, like this is specifically the dust mite allergen. It comes in, it's within the tissues, so it's going to be exposed to dendritic cells, to mast cells. It's going to be taken up by the dendritic cells, and then it's going to load it onto an MHC class 2, and then it's going to present it to T cells, and eventually we're going to get Th2 T cells, because these are the ones that are implicated with your allergic reactions, basically. So the dendritic cell, remember we have signal 1, we have signal 2. So signal 1 is going to be MHC binding to the T cell receptor. Signal 2 is going to be B7 and CD28, right? And then signal 3, it's not a real signal, but signal 3 is like the cytokines that the antigen presenting cell is giving to the T cell. It's like, hey, I need you to become a Th2 cell. So what it's going to do is it's going to send it IL-4, IL-5, IL-13. And this is what's going to allow it to prime or like become a Th2 T cell. Okay, so now we have a Th2 T cell. Now it's going to go do T-dependent B cell activation. So the Th2 cell is going to go to the B cell and be like, hey, we need IgE. We have like a really bad parasite allergen, which of course it's just dust mite, but that's what the body's thinking. So it's like, hey, we need some IgE. So it's going to send IL-4 to it to make it, and we get signal 1, we get signal 2, which in this case is going to be CD40 and CD40 ligand. And then the signal 3, IL-4 is going to be given to it. Once the B cell is completely activated, it turns into plasma cells, you get memory B cells, and then now we have all this anti-dust mite allergen IgE being made. So now plasma cell goes back and it's again producing more of these antibodies, and then that's what kind of ends the sensitization. It's not doing anything yet because this takes a little bit of time to happen. But now it's going to stick in the surface of the mast cells. So now here's a mast cell. It's going to bind to the Fc epsilon, like the Fc portion of the IgE antibody with its Fc epsilon receptor. And now the second time we see it, that subsequent exposure, as soon as you see this dust mite antigen, we get mast cell cross-linking of that IgE, and then it's going to completely degranulate and it's going to release histamine. Another thing that's located within the granules is tryptase. I don't know why I bolded that. Maybe that was like a question. I don't remember. But histamine's, of course, the main one. This is why we take antihistamines. But I guess also no tryptase is involved. Not sure, but this is what's causing your allergy symptoms. And again, it's really important to understand it's on that subsequent exposure. So what are some symptoms of like, mast cell degranulation? Again, just think allergies, honestly, but there's different types of allergies. Like your seasonal allergies, that's kind of where we think this. Like your eyes get teary, you're coughing, you're sneezing. Sometimes people are wheezing if it's really bad. Your nasal passages get really swollen, all sorts of stuff. Just think of typical allergies. But some people get allergies, they have really bad GI upset. So this is like, histamine can also affect your GI tract. So you can get diarrhea, you can get vomiting, and again, it's going to depend. And notably, mast cell degranulation, histamine, and we talked about this with inflammation, is it's going to increase the diameter of your blood vessels. And again, it's thinking that we need like an inflammatory response. So it's thinking that, okay, let me increase the blood flow to the area so more white cells can come to the area and get rid of it. But this is really bad, and this is what leads to allergies killing people, is if it leads to anaphylactic shock. So anaphylactic shock is basically when the blood pressure drops so much that now, because the blood vessels are so dilated, that you can't get blood to the tissues. Okay, so it's like, I don't know if you guys covered shock and cardio block, but this is like distributive shock because it's an issue with distributing. It's like your warm shock because think of all the vasodilation happening with the blood vessels. So how this looks is, this is kind of confusing, but I'm gonna try and explain it with how they would ask a question about it. So it is immediate. So what you're gonna see immediately is what's known as a wheel and flare. So a wheel and flare looks like this. And if you've ever been like skin tested for allergies, just they like inject a little allergen under your skin. This is what it looks like. It's just mast cell degranulation within that area. So think of it as like a really tiny inflammatory response. So it's gonna get big, it's gonna get really itchy, and so forth. But what happens, and that's kind of what it's showing here, is like this is a peak expiratory flow rate. So you can think of this as being like an asthma patient, right? So if they were to blow out, so they're expiring, here's your antigen. Immediately what you're going to have is you're going to see an immediate reaction, and then about 6-ish hours later, I know that's not great, but that's about what happens, like 6-ish hours later. At this point, we're recruiting the other inflammatory cells to the area. So like we're recruiting macrophages. Again, think about that's why histamine's vasodilating, so we can get these other white cells to the area. We're trying to get these inflammatory cells to the area. Then you have late phase, you're going to see what's known as an induration. This is what an induration is. It's basically just like a very thick, hard, like version of the wheel and flare. So it looks like this. It's hot to the touch. Again, it's like the inflammatory response, but like happening a little bit bigger scale now. But it's going to be in that same area, and it's going to be 6-ish hours later, which is just like an annoying fact you have to remember about the immediate type hypersensitivity. You're still kind of seeing symptoms 6 hours later. Does that make sense? So first you're going to see wheel and flare, and then you're going to see an induration. Okay, that's type 1. Good? Allergies, we love allergies. Okay. So type 2 hypersensitivity, so now we're in A, B, C, D. So type 2 is going to be B, so this is antibody mediated. This is going to be, think of this as being an antibody against a stationary antigen. So it can be a receptor, it can be some sort of antigen that's on the surface of something. Again, the important thing is they're not complex. We're not making antigen antibody complexes that are circulating around. Antibodies are binding to antigen, but they're not, the antigen antibody complex itself is not circulating around. And I'm emphasizing this because there's different types of type 2s, and they can get kind of confusing. You just kind of have to remember, like, it's not like the complex in type 3 is what's causing the issue. Here, it's the actual antibody binding to the antigen. And hopefully if you see an example, it'll help. But so there's different types of type 2s, and this is what I was kind of saying. So you can either think of this as destroying cells, making the cells dysfunctional, or causing inflammation. So I'm going to start with the cell dysfunction because I think this is the best example of a type 2. You guys kind of had it maybe a little bit with neuro. So cell dysfunction, basically think of the antibodies binding to cell surface receptors. Again, surface. It's a surface receptor. It's on the surface of something. It's stationary. It's not moving. This can either block the receptor or it can activate the receptor. So the effector in this instance is the actual antibody. It's what's causing the dysfunction. So 2 classic examples, myasthenia gravis and Graves' disease. Myasthenia gravis, when the antibody binds to the receptor, you're blocking this function. And that's why with myasthenia gravis, you don't have function of the nicotinic receptors in the neuromuscular junction. That's why symptoms are like over time it gets worse because it's like they can't keep up. Graves, however, is it's an activating binding. So when the antibody binds to the the TH4 receptor, the thyroid hormone T4, that's the one. The receptor for T4, or TSH? I think it's TSH. When it binds to the TSH receptor, it's going to cause more TSH to be released. So now we have more T4 being released, and now all this T4 and everything is going to like cause the symptoms of Graves' disease. So you see like the like the eyes bulge out because again, you have antibodies targeted against that receptor. Okay, so both of these are examples of a type 2 hypersensitivity in terms of causing dysfunction. It can either block the receptor or activate the receptor. For cell destruction, this is kind of like, think about like the antibody itself thinks it's recognizing an antigen on a cell. So I'm going to slide down just to show you an example, like a red cell. sometimes the antibodies think that there's an antigen on the red cell. A lot of the times, just a little hint, it's usually drugs. So if you take a certain drug like penicillin, it's going to like coat the red cell, and then the antibody is going to bind to it. It recognizes it as foreign, and then it's going to destroy it. And now all the red cells are going to basically get destroyed. So that's kind of what we're going to talk about as like a hemolytic anemia. This is what happens with like transplant rejections, hemolytic disease of the fetus and newborn. This one's really, really heavily tested. But another thing with this is it's just, I mean, you're like, how do you destroy the red cell? It's just an antibody binding to it. It's not like the T cells are coming in. But look at the effector cells. You can have complement getting activated. You can get the phagocytes recognizing the Fc gamma receptor binding to it. You can have NK cells binding to it and doing ADCC on it. So it's like you can still destroy these cells even though antibodies are just binding to it. And then lastly, this is like a wide range of stuff, but just the antibodies binding to the cell surface receptor, you can activate complement. And in this instance, this is what happens with like things like Goodpasture syndrome, rheumatic fever. So again, Goodpasture. I don't know if you guys would have covered that yet, but maybe you have. But it's like, think like renal, like it's the basement membrane within like the kidneys. It's also in the lungs. And then rheumatic fever, and which is like in the heart, and then transplant rejections can also do this. Sorry, question. Yes, very good question. So we're going to actually talk about that. So the question was, in that case, is penicillin acting as a hapten, like in this example? And that's exactly what's happening. Penicillin's acting as a hapten. So we're going to talk about that later. Yeah, very good observation. Okay, so we're going to start with this. So cell destruction. So something, it's not always penicillin, but something is acting as a hapten. And then what happens is it kind of binds to like the red cells, and then this causes your body to just produce antibodies against everything. And some people are like, well, if you're just making an antibody against penicillin, why are you still getting anti-red cell antibodies? And remember when we talked about haptens, you get 3 types of antibodies. We're going to review them. But when you get haptens, you get an antibody against the hapten itself. So in this case, it would be penicillin. You get one against the carrier, which in this case would be the red cell. And then you get one against like the whole complex, the hapten carrier complex. So that's why you're still able to like destroy the red cells in that case, even though it's like not all the red cells have penicillin on it. And then there's different types of hemolytic anemias. So when I was specifically mentioning penicillin, this is your drug-induced hemolytic anemia. It's kind of exactly what I said. Penicillin's acting as a hapten, red cells acting as a carrier. You're destroying all the red cells. ABO mismatch, this is blood typing. So if you have type A blood and you're accidentally given type B, that's horrible and that causes cell destruction like immediately. And then hemolytic disease of the fetus and newborn. These are 3 examples of like hemolytic anemias in just overall meaning like cell destruction, like you're destroying the cells. So drug-induced. So we're going to talk about drug-induced. Again, this is the type 2 with the cell destruction aspect of your type 2s. So keep in mind, again, it's like you have cell destruction, dysfunction, and then inflammation with the type 2. So there's a wide range of different types of type 2s. Whereas type 1, it's like allergies. That's it. Allergies and asthma. So that's why type 2s are a little trickier. So I try to put in the corner like what type of type 2 I'm talking about. So in this case, it's cell destruction. So the whole idea behind this is that whatever drug you're injecting is acting as a hapten. So it's going to be injected. Remember, you make 3 types of antibodies when you inject a hapten. You make 1 to the carrier, 1 to the hapten, and then 1 to the hapten carrier conjugate. And this is kind of showing the same thing. So here's your red cell. This circle is the drug, and now you're going to make these complexes that can activate complement, that can get phagocytosed, whatever. You're going to get various types of antibodies made. And this is the bad one. This is the one where you're making an antibody against a red cell self antigen. But this is just what happens when you have haptens being injected. Sorry, questions. Is HDFN the same as hydrops fetalis? Yes, hydrops fetalis is HDFN. So if you learned about hydrops fetalis already, that's what HDFN is. Yeah, that the names they're used interchangeably. So if you have notes on hydrops fetalis, you can like literally go back and be like, okay, this is also HDFN. Okay, so that was a drug acting as a hapten, which can cause destruction. This is ABO mismatch and HDFN. So we are going to go into blood typing a little bit. They do expect you to know how to blood type. So we're going to go into this. So basics on blood typing. If you're type A, so if you're type A blood, this means, and I have figures to kind of help you understand this, but you have A antigen on your red cell surface. That's what makes it type A. So here's your red cell. You have A antigen all everywhere here. A weird thing about blood types is it doesn't follow normal like antibody production. You don't need sensitization. You automatically make anti-B. And it's just you don't have to know like the why this is a thing. Just know you automatically make anti-B antibody just out here in the serum. So your blood has A antigen on it, and then you make automatically anti-B antibody just floating around in the serum. If you're type B, it's the exact opposite. You have B on the red cell, and then you make anti-A automatically, just floating around in the serum. Again, you do not need sensitization. You do not need to be exposed to type A blood, and then you make a like anti-A antibody. It happens automatically. If you're type AB, you have both A and B on your surface. You don't make any type of antibody because you have both of them on your surface. It would just destroy your own cells. That would be counterintuitive. And then if you're type O, the O is actually supposed to be 0. There's absolutely no antigens on the surface. So what do you automatically make? You automatically make anti-A, and you automatically make anti-B antibodies that are just kind of floating around in the serum. Again, do not need any sensitization whatsoever with this. So here's kind of what I tried to draw, but this is prettier. So if you're type A antigen, anti-B, automatically. If you're type B, you have B antigen, anti-A antibody, automatically. Type AB, you have A and B antigen, no antibodies, because again, it would just attack your own cells. And if you're type O, you have 0 or O antigens on the red cell surface, but you have both anti-A and anti-B in the serum. And again, you don't need sensitization. So how are we tying this into hypersensitivity? So remember that type 2 hypersensitivities are when an antibody attacks a surface antigen. So how would this make sense with the type 2 hypersensitivity? Well, what happens if we give type B blood to someone who's type A? So here is type B blood. We're giving it to our type A person who, look at that, has anti-B antibody in its serum. So this is going to be exposed. It's going to be exposed. Here's your type 2 hypersensitivity. The antibody is going to bind to the surface, and then you're going to get lysis. You're going to get complement activation, phagocytosis. You can do ADCC with this. So you can get all sorts of issues that can cause lysis of the red cells. Okay. So this is kind of what we talked about, and now it's just like fun facts. Type AB is your universal acceptor because they don't have anti-A or anti-B antibodies. They have no antibodies, so technically you can give them whatever blood type you want because they don't have antibodies already made, and they will not make antibodies because if they do, they're going to attack their own blood. So the body won't let it do that, basically. So that's why universal acceptor, you can give it whatever type of blood you want. O is the universal donor because this has no antigens on its surface, just absolutely none. So the anti-B, if you give it to a type A person, the anti-B antibodies have nothing to attack on it. If you give it to a type B person, the anti-A antibodies have nothing to attack on it. So this is kind of why it's like, okay, AB is your universal acceptor, and then O is your universal donor because of this like idea of the antibodies and the antigens. Okay, so that's A and B. Okay, so A and B antigens are on their own. So you're automatically, again, just really keep this in mind, you're automatically making this without sensitization. So again, that's only for A and B. There's also something known as Rh factor, which sometimes it's referred to as D antigen, and I'm going to refer to it as D antigen because I think it's helpful to think of it as like an antigen. So this is Rh. So if you're A positive, that means you're type A Rh positive. If you're A negative, you're type A Rh negative. So Rh is talking about this D antigen. So do you have D on the surface? So if you were A positive, you would have A and you would have D. You would look like this. So like this is A pos. Okay. So think of Rh as another antigen, D antigen, in addition to your A and B antigens. So kind of like what I drew here. If you're A pos, you have D. If you're A neg, you don't have D. But difference, star this, difference from ABO is if you're D negative. So let's say you're A negative. Let's say you're A negative. So you're going to have A antigens on your surface. You're going to have anti-B out here because that happens automatically. But you don't have D antigen on the surface. You do not automatically make anti-D. You don't do this. You don't have, okay, if I'm D negative here, I make anti-D. That does not happen. You do not make it automatically. It's different from like ABO typing. Okay, so with D antibody production, anti-D antibody production, this absolutely requires sensitization. You need to be exposed. It's kind of, think about like the mast cell thing again. So if you, the first time you eat a peanut, nothing's going to happen. The second time you eat a peanut, if you made antibodies against it, that's when you're going to have an issue. And it's going to be the same thing here. If the first time you're exposed to D blood, let's say, not a big deal. But now you made anti-D, like after that first time, okay? So this is kind of, oh, that looks like a P. And we're going to talk about this, but this is the biggest principle behind HDFN, hydrops fetalis, whatever you want to call it. Okay, so that's like a really big thing to understand is you do not automatically make anti-D. So I added the D antigen so you can kind of see visually what's going on. So if you're A positive, again, like type A, Rh positive, you have the D antigen as well as A, you automatically make anti-B. If you're type A negative, you only have A and you only have anti-B, okay? And then so forth and so on. So the true universal acceptor, so now we can kind of update our thinking, right? So the true universal acceptor is O negative. So I kind of lied to you by saying type O is universal acceptor. It's technically O negative, and that's why. If you're O negative, you don't have this D antigen on the surface. So even if you were to be exposed to, let's say, D positive blood, you'll never make anti-D if you're O negative. Donor. Oh, yep. Good catch. I was like, what is she talking about? Good catch. Yes, very good. Donor. Universal acceptor is AB positive. Yes, they always say I need O-neg in the doctor shows. That's why. It's because of this. It's because this is the true universal donor. Thank you. Good catch. I'll change that on before I send it to Sarah. Yeah, so this can be donated to anything because, again, there's nothing on the surface whatsoever. Whereas O positive, there's D on the surface, like there is D on the surface. So if you can't give it to someone that doesn't have D on the surface, okay? So O neg can only receive from O neg people though, because if you go give O pos to an O neg person, that's their sensitization event. That's, okay, now I've been exposed to this antigen, I'm gonna make anti-D antibodies now. So the true universal acceptor is AB positive. So has A antigen, has B antigen, and has D antigen. So this means AB positive, since it's the true universal acceptor, can literally receive blood from any person. It already has D antigen, so even if you give D positive blood, if you give D negative blood, it doesn't matter. It's not going to make anti-D because it would attack its own cells. So that'll never happen. So that's why it's like the true universal acceptor. And then this is kind of like going back into this, but you don't have to memorize it. You can just kind of think about it. So if your blood type is AB positive, you can receive from any type, right? So this is your universal acceptor. And again, it's AB positive. If you're O negative, so if your blood type is O negative, you can receive from literally, how did they do this? Yeah, so they did this backwards. So if you're O negative, you can receive from literally any person. Literally any person. But if you're AB negative, A negative, B negative, you have to think about both D antigen and like the other two antigens, A and B. So for example, A and B, remember I said you can get blood from an A person, you can get blood from a B person, you can get blood from an AB person. But since we're negative, you can't get blood from someone who's positive. So all of these, with the exception of O neg, which is given to everyone, you can get from B neg, A neg, or AB neg. If you're A neg, you can only get from A neg and O neg. B, same thing. B neg, same thing. O neg, again, can only get from O neg. That's it. But the positives, they kind of have an advantage. So if you have Rh positive, you're kind of at an advantage because you can get now from both positive people and from negative people. You can get from positive people because they're the same blood type, right? They have D antigen clearly, so it doesn't matter, so do you. So that's fine. But you can also get from negative people because even if you don't have D antigen, it's fine. It's not like anti-D already exists. They're not automatically making that. Okay? Makes sense? We love blood typing. Okay, so on my exam, they asked like if you can identify who the dad is by using blood typing. So we're gonna do quick genetics, nice little punnett squares. So something to know about ABO antigens, they're co-dominant. So if mom's A and dad's B, you're gonna be both of them will be expressed. It's not like normal genetics where it's like one's dominant, one's recessive. It's like this is like both of them are dominant. So if you're type A and dad's type B, then you have the potential of having both A and B. So if they're there, they'll both be expressed. But of course, D antigen likes to be its own thing. D antigen is not co-dominant. So this follows your normal dominant recessive genetics. And I'll kind of give you an example of this. So here's ABO. So ABO, remember, this is your co-dominant expression. So a lot of the times when they say that like mom is type A, it's going to be one allele is going to be like a big A, and the second allele is going to be like an I. And it just means that there's nothing there. It's A and then an I. Like this means nothing. Dad, he's type B, so he's going to be B and an I. And again, the I means nothing. And then you do your Punnett square. So we have A and I. I tried to color code it so you can see where I'm getting the letters from. So you can see this person, if you have mom's A and dad's B, you have a 25% chance of being type A, 25% chance of being type O. So again, type O means that there's nothing on the surface. So you would need this one from mom and this one from dad. 25% of being AB and then 25% of being B. So that's just for ABO. Can you be big A instead of A little? No, you can't. Good question. You can't. And that's a good thing to remember because if like you're like again, mom's A, dad's B, and your baby's O, it's not like, oh, dad can't be the dad, right? Like, yeah, so it's usually gonna be this. Big A, I, and then the big B and then I. And that's how you get a type O. But the Rh antigen follows its own genetics. It kind of does the Mendelian genetics. So what this means is if you're D positive, so Rh positive, this now can be big D, or big D, little d. Nope, that's an I. Big D, little d. So this, again, would be dominant. So this is dominant. So this means D would be expressed if you have a big D. Here, this D would be expressed because the big D is dominant to the little d. The only way you would be negative from this instance, like Rh negative, is if you have two little d's. So here's an example. I kind of gave you both scenarios. So like let's say mom was Rh positive with the two big D alleles and dad is Rh negative. Rh negative can only be these two little d's. That's the only way you can have Rh negative. So again, you can see 100% the baby's going to be Rh positive if mom was big D, big D. Will they tell you? I don't think they will, but I think it's still going to be fine. Like you can still figure it out, basically. Yeah, I'll see if I can find a question, but it's not going to be an issue. A lot of the times it's more of like something like this. But the big takeaway is like if mom is positive and dad is positive, your baby cannot be negative. There's no way it can be negative. Okay? I mean, sorry, I said that opposite. If mom's positive and dad's positive, you still have a chance that the baby can be negative because of this concept of the big D and the little d. If dad was big D and little d, then you have like a 25% chance of being little d, little d. So it's like just more of something to keep an eye out for, like not to just rule out immediately. If mom's positive and dad's positive, the baby can technically still be negative. So again, here's your Punnett square, fill it out. So you can see 50% of the time the baby's going to be Rh positive, the other 50% of the time the baby's going to be Rh negative. Okay, so this is just an example of like how you can use blood typing to figure it out. So again, when you see this, you'd be like, okay, mom's A, so mom's this, dad's B, so dad's this, and then the RH is going to be a little bit trickier. So mom can either be big D, big D, big D, little d, but then dad can only be little d, little d. So that's pretty much how you would set it up. And then that's not what they're gonna like. They would have to give you more. They can't just you can't just assume it's gonna be one or the other. So they would have to. I don't think they'll tell you, but it'll be something where you don't have to. It's not gonna make or break knowing that information. Okay. So implications of this. So this is what I was trying to say. Mom could be Rh negative, but then you can have a baby who has the D antigen. So if mom's D negative, but dad's D positive, you can have a baby that has D antigen. So don't you see that can't this be an issue, right? So if let's say mom is sensitized to D antigen because she's literally carrying a baby that has D antigen, so she gets sensitized from this somehow, the anti-D antibody is IgG. This is the only antibody that can cross the placenta. And this is kind of where HDFN gets confusing. So let me know if you guys have questions about this, but we're going to talk about it for the next several slides. So anti-D antibody, when you make anti-D, this is an IgG antibody. IgG can cross the placenta. So this can be an issue if mom gets sensitized to D antigen, makes anti-D, and then is literally carrying a baby who has D antigen, right? So this can be an issue. So this is going to be kind of like you have to think about this if you have any mom that's Rh negative. You have to think of HDFN. If you're Rh positive, guess what? You will never have to worry about HDFN. So if you're type A positive, AB positive, B positive, you don't have to worry about this ever. It's like good for you, right? It's like, yay. But if you're Rh negative, you will absolutely have to worry about this in the future. So if mom's Rh negative, so A neg, AB neg, O neg, B neg, and she's sensitized to D antigen from her baby. So again, this is like any type of bleed during the pregnancy. Really common to have like little placental bleeds during your pregnancy. I mean, the baby's growing, things are happening, you're moving around. You can get like tiny placental bleeds. It's not a make or break situation. A lot of the times, this is what happens. You get like these little baby microbleeds. Well, baby, like I just mean like tiny microbleeds within the placenta, which is going to sensitize mom. So that's going to sensitize mom. So now mom is making anti-D antibody, which is IgG. But this is going to be an issue for her next pregnancy if she has another baby that's Rh positive. And I'm going to show you pictures because you're like, wait, I was following until the last part. It's usually what happens, but I'll show you pictures. But this is an issue, like if it were to cross the placenta and attack her baby during the second pregnancy, that's called HDFN. So that's called hemolytic disease of the fetus and newborn. That's what causes hydrops fetalis. There's other causes of hydrops fetalis. When you guys do micro this summer, you'll see like there's some viruses that actually can cause hydrops fetalis, but it does so without causing HDFN. So it's kind of like HDFN and hydrops fetalis are like the same because that's what HDFN causes. But just keep in mind you can have other causes of hydrops fetalis. And then how you protect against this, because you don't tell Rh negative moms to never have children, don't have more than one kid, we have something, it's called RhoGAM. And RhoGAM is basically like Rh immunoglobulin, RHIG. So we're going to go through it more. So again, pretty sure your mom had to take RhoGAM. Yeah, so if your mom is O neg, A neg, B neg, AB neg, she 100% took RhoGAM. It's just, we do it now. I'm like talking as if I'm a doctor, right? Like the doctors do it now automatic. Like if you're Rh neg, you're getting RhoGAM. They're like, I'm not even gonna risk you getting this microbleed during your first pregnancy. And even if you're like, well, I swear I'm only gonna have one pregnancy, they're like, I don't care, you're getting RhoGAM. And we'll talk about RhoGAM. So mom has to be Rh negative. Dad has to be Rh positive. If dad's Rh negative, this is not an issue. So if mom's Rh negative, dad's Rh negative, the baby can only be Rh negative. Remember, Rh are these little d, the little d. This is all it can be. So if dad's Rh neg, mom's Rh neg, the only possible combination for the baby you can get is little d. So dad has to be Rh positive, mom has to be Rh negative. So this means the baby can be either or, and they don't risk it. They're like, we're not going to risk that it's going to be this. Like, it's a 50% chance, we're not risking it. So that's why these people get ROGAM regardless. So first pregnancy, this is your sensitization event, right? Again, think about peanuts. So you have to, the first time you have a peanut, yeah, it's like your body's clearly having an allergy. It's going to make an IgE antibodies against it, but nothing happens that first time you have a peanut. Same thing here. The first pregnancy, nothing happens. You get sensitized. This is where mom gets sensitized. So mom's Rh negative. Now let's say her baby's Rh positive, and now she's sensitized. We have like a little microbleed, something happens. So mom's going to be experiencing this Rh positive blood. So now her blood is like, oh, here, I'm being sensitized to it. I'm going to make anti-Rh or anti-D antibodies. So now mom has it. Does nothing to the first baby. Does absolutely nothing to this baby. And the reason why it does nothing to the baby for the first one is because when this sensitization event happens is usually close to the third trimester or like very close to when she's about to deliver. Delivery is very traumatic, so you're definitely getting mixing of the blood during delivery. So that's when mom's developing it. So it really doesn't cause issues at all for the first pregnancy. The second pregnancy is where it's an issue. Again, think of sensitization and destruction with the peanut again. Think for sensitization, degranulation. So mom, she's still Rh negative, right? So she's still Rh negative. Dad's still going to be Rh positive. Maybe. I mean, maybe it's another dad, but let's say the same dad. He's going to be Rh positive. The baby's going to be Rh positive. They're assuming. They're like, all right, we're going to just assume baby's going to be Rh positive. Now what can happen is mom's anti-D antibodies that she made from the first pregnancy immediately can cross the placenta because they're IgG antibodies, immediately can cross the placenta and kill the baby literally while it's in her stomach, which is known as in utero, when it's still within the body. That's hydrops fetalis. So what they do is they give ROGAM during every pregnancy. Every pregnancy, if you're Rh negative, they don't even care. They don't even want to know what the dad is. They're like, we don't care. If you're Rh negative, you're getting ROGAM. And the reason why is because it's going to even just prevent sensitization. And we'll talk about what ROGAM does, but it basically just like coats, it's like just coating the red cells. So it's like the red cells can't see anything. I'll show you. So this is erythroblastosis fetalis. So this is hydrops fetalis, HDFN. It's all the same thing. So this is what happens without ROGAM. So ROGAM's over here on the right. So here's your placenta. So again, here's your maternal circulation. So let's say mom, remember, she's D negative. So mom does not have D antigen on her red cells. This baby is D positive. So they're showing like a biconcave red cell with the little blue circles on it. This is going to be your D antigen. So this is your D positive. Okay. Now we deliver, again, very traumatic experience, even like just microbleeds throughout the pregnancy. It doesn't have to be delivery, whatever, but you can get mixing of the fetal blood into the maternal circulation, which is what they're showing here. So here's your fetal blood mixing with the maternal circulation. Mom is D negative. So basically what happens is she makes antibodies. She literally is going to make antibodies against the D antigen. Again, not an issue for the first time. And remember, every single time we get B cell activation, we get plasma cells and we get memory cells. Okay. So again, first pregnancy, not an issue, literally not a big deal. But now we have memory cells. We have more specific, they can be more specific the next time. Here's your second pregnancy. Mom's still D negative. Baby's now D positive. So now as soon as this happens, you don't even really need mixing. You don't even need this mixing to happen. The memory cells are going to remember. It's going to be like, oh, we saw this already. It's bad. Here's IgG, like anti-D IgG, that can cross the placenta and it can basically attack this baby. So they're showing the antibodies binding to the antigen, attack this baby, and then that's what causes hydrops fetalis, erythroblastosis fetalis, HDFN. This whole process is known as hemolytic disease of the fetus and newborn. So it's a hemolytic disease because we're lysing basically every single one of the red cells within the baby. But you prevent this with ROGAM. And ROGAM is basically it is an antibody. It's basically an antibody. And what it does is it's IgG antibody. And it coats, like I said, it kind of coats the red cells. So when this were to even cross the placenta, so if, again, this is in the baby. This is your baby's red cell with D antigen on the surface, the blue circles. Even if this crosses the placenta to get to mom, since it has ROGAM on its surface, which is coating all of the D antigens, when it gets to mom, mom doesn't see it. Like she can't see it because it's covered. Remember, a function of antibodies is neutralization, right? So you can think of it as like these ROGAM antibodies are like neutralizing the effects of this. So here's your red cell, the effects of this D antigen. It's completely neutralizing it. So mom's immune system doesn't see it. Mom's immune system doesn't get B cell activation. You don't get memory cells. You don't get plasma cells. You don't make anti-D antibodies. And it has significantly decreased the incidence of HDFN to the point, this is again why they give it to every single Rh negative mom. And ROGAM, it's actually a shot in the butt. So that's another thing to look forward to if you're a D negative person. You get to look forward to that. Yeah, you're gonna be like experiencing everyone's blood type. Be like, Mom, what blood type are you? What am I? Yeah, actually, I had a lot of friends when we learned about this. A lot of friends that like typed their own blood. They're like, I don't know if I need ROGAM. Like I don't know. But you can also do it just asking your mom and dad what type they are. You can kind of figure it out. Okay, so another version of HDFN. So remember, sometimes, and again, this is like usually during the course of a pregnancy, if the baby's D positive, it's gonna get into mom's circulation. It's gonna happen. And again, mom has to be Rh negative. Baby has to be Rh positive. It's gonna get in there. Mom's gonna be sensitized now. She's gonna make antibodies, but it does not affect the first pregnancy. This does not affect do not affect her first pregnancy. If the second pregnancy, if this baby now is Rh positive, mom's still gonna be Rh negative. Now these can immediately cross the placenta and kill this baby, basically. Very sad, but that's HDFN. ROGAM, again, it's going to coat, I don't think they really show it well here, but it's coating these D antigens like this. So even if it gets over here, mom does not make anti-D antibodies. So baby is safe, totally fine. Is it too late to give ROGAM after the first pregnancy? No, you want to give, if, let's say, like, maybe, yeah, your first pregnancy, you didn't have maternal care, it's fine. Just, that's why you get it every single pregnancy. Like, they don't even, like, look at your chart. They don't even care. They're like, you're Rh negative. It's like, oh, I already got ROGAM. Don't care. They're like, we're going to give it to you. Like, you just get it every single pregnancy, basically. And you usually get it, like, towards the end of the trimester, like, towards, like, second, third trimester. Yeah, ROGAM's really cool. When I worked in a blood bank lab, I did a lot of this, like calculating how much ROGAM like patients need. And it's like basically what you do is you get a sample of like the placental blood, you get a sample of it and you test it and you see how many fetal red cells are there. And then the more that are there, the more ROGAM that they get. So it's like a calculated process. You don't ever have to know how to calculate ROGAM unless you're going to work in a blood bank lab. You don't have to know how to do that, but just understand that it's an antibody. It's RHIG. So RH for Rh antigen, Ig. It's literally an antibody, and it just coats the fetal the D antigen on the fetus. That's a good question. So how long does it last? I would say the same length of time as like a normal IgG antibody. So like it lasts for like a little bit, but it's going to wane over time. And again, so in theory, technically you only need ROGAM once, but again, they give it for every single, like they don't even want to risk it. They give it for every single pregnancy. So even if you were pregnant and then you literally got pregnant the day after you delivered, I don't even know if that's possible. I don't think that's possible. But just meaning like you get pregnant really quickly after that first pregnancy. So you're like, oh, but I got ROGAM like a year ago. It's like they don't care. They're like, all right, more ROGAM. Like they don't care because it's like it's too big of a risk. But I would assume it would still be in there actually because IgG does tend to last for a little bit of time. But they're still going to give it to you. And of course, antibodies wane over time. You see that with COVID, right? Okay. All right, so that was cell destruction. How do you guys feel about that? That's always like the trick. I think that's always the tricky part is like blood typing and then HDFN. It's always tricky. Do you guys have questions about that before we kind of fly through the rest of the stuff? And I have some good practice questions on this, so you can kind of see how they would ask this, see like kind of where those gaps in your knowledge are with this. Okay, cell dysfunction. Again, this is more neuro and more like endocrine, but remember this antibody can so this is type 2 hypersensitivity. Yeah, so we're taking a step back. So we just talked about cell destruction. So now let's go back to cell dysfunction. That's the second type of type 2 hypersensitivity you can have. This is where the antibody is going to straight up bind to a stationary antigen and causes it to be whatever that is. Usually it's a receptor, block the receptor or activate the receptor more. So Graves is a good example of activating the receptor. Yeah, so it is TSH. See, I still remember stuff from med school, right? So here is your, let's say this is your thyroid cell. So here's a thyroid cell. Thyroid. Here's no, because this yeah. Oh my God, now I forget. I think it's the thyroid cell. So it has TSH receptor. Yeah, it is the thyroid cell because TSH is made pituitary. Okay. Anyway, so here's your TSH receptor. Normally it binds to TSH, right? So normally TSH comes to it, and then if you need more T3, T4, it's gonna be more TSH. But with this, you have an antibody that's going to literally bind into the receptor pocket. And specifically with Graves' disease, this binding causes more activation. For whatever reason, this TSH receptor thinks that this is like, oh, like TSH is here. Like, let's shoot out tons and tons and tons of T3, T4. And then that's how you get Graves' disease. It's basically like hyperthyroidism, right? With myasthenia gravis, this is where these are the postsynaptic nicotinic receptors within like the neuromuscular junction, right? So that's here. So here, it's kind of the same idea. We have like a basement membrane. We have the nicotinic receptors here. And usually they're going to respond to acetylcholine, right? So acetylcholine is going to be released from the presynaptic neuron. It gets released into the synapse. It binds to it, and then you get muscle movement and whatnot. But in myasthenia gravis, we have an inhibitory receptor. So now we have an nicotinic receptor antibody, but it's not acting how these TSH, like anti-TSH receptor antibodies are acting. It's acting by, blocking this postsynaptic nicotinic receptor. So now you really don't get muscle movement and whatnot. So that's kind of more of like your myasthenia gravis scenario. Okay, and then the third type of the type 2 hypersensitivity reaction. So we did cell destruction, cell dysfunction. Now we have inflammation. It's exactly how it sounds. Just the fact that the antibodies are binding to this stationary antigen, you're getting complement activated, you're getting inflammation activated, because think about all the things that happen when the antibody binds. Again, remember I said NK cells can come in, do ADCC. We can have phagocytic cells coming in, recognizing the Fc gamma portion, binding to it, phagocytosing. And again, we have technically here an antibody binding to an antigen. You can activate complement this way. You can get complement activation. So the two classic examples of this are good pasture and rheumatic fever. You will go into this path second year, like you'll go into what this is. I think POEM you'll cover good pasture, and in renal, especially renal, because you see a lot more glomerular issues with good pasture. But all it is a type 2 hypersensitivity. So even if you were to get this question on a boards exam, right, later next year, and it's just, what's the mechanism behind good pasture? It's literally go back to like this lecture right now and be like, oh, type 2 hypersensitivity. We have an antibody binding to a stationary antigen. And in this case, it's recognizing basement membrane antibodies, so which is going to be in the glomerulus, and then it recognizes similar stuff in the alveoli. Rheumatic fever, this you'll probably learn this summer with micro, a specific type of bacteria. It's actually the strep that causes strep throat. It's going to come in. It's going to, again, you don't have to know, like, do not memorize this slide. You will not be asked about this. But here it's kind of like showing like how you can get activation of antibodies against strep, which is what you want. You do want antibodies against the bacteria that causes strep throat. But interestingly, this part right here, several heart tissue proteins are recognized by these antibodies. So it's like you have cross-reactivity. And they just see this a lot of the time with these anti-strep antibodies that you're making, specifically against like the strep throat, like literally against your strep throat. You're trying to get rid of this strep bacteria, and it just cross-reacts with the valves in the heart. And that's actually what causes rheumatic fever. So you can see this here. It's these same antibodies that we made right here against the strep that are now binding to the myocardium and specifically to the valves. Sorry, question. Do you recommend we know the first aid mnemonics with the paths? I don't remember off the top of my head what all those are. There are like a few that I recommend. I have one that I'm going to show you. But if I haven't had it on here, I don't think you need to worry about it yet. You'll go into it more second year. But afterwards, you can tell me like what pages I'll look, and I'll see exactly what you mean. You're so welcome. Okay. So again, think inflammation, think good posture, think rheumatic fever. Okay, type 3s. So type 3s, I'm not going into like actually like any detail with this because Thursday, we're talking about lupus. So this is like lupus and rheumatoid arthritis. So these are your classic, did I say classic? Type 3 hypersensitivities are lupus and rheumatoid arthritis. So the reason I'm not going into like any detail today is because like we're going to talk about it a lot on Thursday. I said, yes, classic type 3. I really want you to think of lupus and rheumatoid arthritis with type 3s. It's like because it has everything. It has everything you can ever imagine in type 3 hypersensitivity is present with lupus and rheumatoid arthritis. But at the very core of this hypersensitivity reaction, antigen antibody complexes. Like, yes, like I just kind of wanted to be clear with the terminology from the last one. Remember, it's a stationary receptor. So we have maybe like a receptor neuromuscular junction. We have a basement membrane receptor, whatever, or we have a red cell antigen, but it's going to be bound by an antibody. It's not moving. Yes, technically the whole blood cell is moving through the blood. Yes. But when I'm talking about antigen antibody complexes, I'm talking about just straight antigen. If this red cell antigen popped off and now that's what the antibody was recognizing, this would be your antigen antibody complex. So that's what's meant by these antigen antibody complexes. Like you're getting literal complexes. So here's kind of like a good picture of one. This is actually what happens in lupus, which we'll talk about tomorrow. But in lupus, you make antibodies against your own DNA. It's really not great. So this is showing DNA, and then here's antibodies against your DNA. Beautiful. Immune complex. That's what an IC is. So here's your antigen, DNA, and here's your antibody against it. This is like the heart of your type 3s. And the reason why this differs from the type 2s is because now this complex, it deposits somewhere. Guess where it deposits? In rheumatoid arthritis, deposits in the joints. But with lupus, it also kind of deposits in the joints. And that's why if you know anyone that has lupus or you've ever like seen, if you're familiar with lupus, they also have joint pain. And it's the core is because of these antigen antibody complexes that circulate, boom, deposit somewhere. And they cause these symptoms. Okay. So again, I don't go into like lupus and RA. We're going to talk about that on Thursday, but I did want to talk about these two reactions down here, classically asked. Sorry, I keep saying classically now, but classically asked with exam questions because they're tricky. And you only see them in type 3s. So anyway, type 3, this is your IgG antibody. It's a soluble antigen. So again, it is not stationary. It is not stationed on something. So it's a soluble antigen. It's out and about. So again, antigen antibody complex is great at activating complement. Excellent at activating complement. So we get complement activation. We get phagocytes that think that they need to gobble this up and take it to the spleen so the spleen can take it out. So we have all sorts of stuff happening here with the type 3s. So again, this is kind of like your main effector with the type 3s is complement. That's what's causing all this inflammation and everything. So here's your immune complex. Antigen are these dots. I don't know if you can see it, but here are the antigens, these dots, and then there's 2 antibodies per 2 dots, basically. And that's your complex. So we have 3 immune complexes here, and then they deposit. And 2 things that you can see classically with type 3s are known as serum sickness and the Arthus reaction. Serum sickness, think general, like it's a generalized, and I'll talk about what they are. But then think Arthus reactions are localized. Arthus reactions actually look very similar to the, remember after the wheel and flare, you get the induration, like late phase induration with the type 1. Arthus reactions actually look very similar to indurations, but we'll talk about how you kind of differentiate them. Okay, so serum sickness. So serum sickness, it tells you serum sickness. It's in the serum. It's general. It's in the whole body. So it's like generalized. It's like everywhere in the body. Again, it's a type 3 hypersensitivity. These are only terms associated with type 3 hypersensitivities. Generalized is how you differentiate serum sickness from Arthus. This occurs 1 to 2 weeks after exposure. Do not get confused with type 4s. I know we haven't talked about type 4s yet, but type 4s are your delayed type hypersensitivity, so they usually occur around 1 to 2 weeks after exposure. Don't get confused. There's going to be like ways that you can differentiate between these two. And basically all serum sickness is you're getting antigen antibody complexes and they're depositing everywhere. So this is an example of serum sickness. These are, and all these little dots are antigen antibody complexes like depositing in the skin, and you're getting like this systemic reaction. Arthus reaction is a localized version of a type 3 hypersensitivity reaction. It's going to occur 4 to 10 hours after exposure. Okay, so you're going to be exposed to something. About 4 to 10 hours later, you're going to get this localized reaction exactly where the injection site was. So if you were injected with something, clearly you made antibodies against it like right away, or you've had previous antibodies to it, and you're going to get this big reaction. And it's localized versus systemic with serum sickness. And again, I'll kind of show you how you can differentiate this then with something like an induration with the type 1s. So here's kind of like the charts for serum sickness. So some drugs can cause serum sickness, especially those that act as haptens. So penicillins can do this. So penicillins are famous for causing type 3s. So we did talk about how penicillin can act as a hapten and then absorb onto the red cell membrane. And then in this instance, it can cause a type 2, right? Drug-induced hemolytic anemia. But rule of thumb is if penicillin was given and then like either you get this type of reaction or you get this type of reaction after being given like a penicillin, it's like they classically cause type 3s. Okay. So here's kind of what's happening. So here's level in plasma. So this is the patient's blood. We're going to inject a foreign antigen here. So it's going to be really, really high in the plasma right away, right? So this is the antigen. Yellow is the antigen. Eventually it's going to wane over time, and then you're going to start making antibodies against it. So you're going to make antibodies against it. And then right here in this blue, this is where you have both antibodies against the yellow and you still have antigens present in the blood. So you're making antibodies, which is what you want, but the antigen didn't wane off yet. So you're getting these complexes being formed. And this is how it's presenting. This is serum sickness: fever, vasculitis, arthritis, nephritis. You can get skin finding. Just think like just generalized just sickness. That's serum sickness, and it's because you injected something into the serum and now it made you sick. But again, it's happening 1 to 2 weeks after you get something. You get injected with something. And then note the x-axis is time in days. So again, this is many days. This is 1 to 2 weeks like after. Arthus reaction, on the other hand, is basically like you get locally injected antigen and locally injected antigen in immune individual with IgG. Okay, so this is saying you're injecting an antigen into someone that already has the antibodies made. So this is someone that's already been sensitized to this red antigen. They already have this. So instead of like causing mast cell degranulation in this instance, since they're IgG antibodies, we're going to get immune complexes forming. Again, this doesn't happen in everyone. It's just like something you can see happen in some patients. Then from here, you can also get activation of mast cells, Fc gamma. Okay, don't worry about that too much. Yes, mast cells also do have Fc gamma, but Fc epsilon is the more important one. But anyway, you're getting this local inflammation, fluid. You're basically getting an induration. Like you're basically getting one. Okay, and it's, again, just think of this 4 to 10 hours after exposure versus this. This is the one that they love to ask about these two. So if when in doubt, think serum sickness or Arthus. That's my professional advice. Well, my non-professional professional advice. Okay, but it's, again, you're going to get symptoms of it, the inflammatory response: increased fluid release, inflammation, blah, 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 all that stuff. So serum sickness is not sensitive. Right, yes. So serum sickness, you need time to make the antibodies. That's why it's about 1 to 2 weeks later. And then, but the antigen's not gone yet. You need the antibodies to get rid of the antigens. You kind of need, like, it's kind of like a backwards thinking. And then in some people, for whatever reason, they get these complexes rather than like the antibodies neutralizing the antigen and getting rid of it. You just get antigen antibody complexes instead. Versus Arthus, yes, you already have these preformed IgG antibodies that are made. And again, instead, it's kind of the same idea. Instead of neutralizing like it should, you get these circulating complexes that can go and just go wreak havoc, cause inflammation, deposit places, and then you get this big induration. Like I wrote here, you get this really big induration with the Arthus reaction. So with Arthus, do you need, no, so you don't, yes, you do need a sensitization event with an Arthus reaction. Yep, so this was made from that's why like the Arthus reaction and the type 1 hypersensitivity like induration are really similar. They're really, really similar. And if you're ever like, again, we'll talk about, I'm trying to not give too much away with the questions, but I'll kind of show you what to look for to know that they're talking about an Arthus reaction. Okay, so this is the mnemonic. I don't know if this is what you were talking about as being in first aid, but this one I think is great. So this is all the examples of type 3 hypersensitivities. So type 3 hypersensitivities are sharp. So the S is for lupus, well, SLE, which is lupus. So systemic lupus erythematosus, serum sickness, hypersensitivity pneumonitis, you'll do second year. Arthus for the Arthus reaction. Rheumatoid arthritis, we're talking about these guys on Thursday. And then post-strep glomerulonephritis. I don't have this, I don't think, in your notes. I kind of added it because you are going to do this second year, but it's really easy to identify in questions. Super, super easy. If they tell you that the patient has streptolysin O antibodies, that's an automatic thing for that they had this strep at some like in some way in the past. Like when they had strep throat, they made streptolysin O antibodies, and now they're having kidney symptoms. That's called post-strep glomerulonephritis, and it's due to these circulating antigen antibody complexes depositing in the kidney. So I just wanted to add it there because it's a really easy, it's almost like a really easy one to kind of identify with when doing questions. Again, I'm always thinking of things that they can put on there that might be you haven't even covered yet. Okay, and then the last type of hypersensitivity, these are your type 4s. These are your T-cell mediated ones. So classic examples of your type 4 hypersensitivities or your delayed type hypersensitivities, so delayed T-cell mediated, any of these. These are going to be like your poison plants, so poison ivy, poison oak, poison sumac. They do that, they cause type 4 hypersensitivities. And then actually nickel in jewelry, so like jewelry, watches, things like that, they can actually cause type 4 hypersensitivities. Those are like the best examples. Okay. So delayed type and then also contact hypersensitivity is like an example of a type 4. So I just think of them as like the same thing. But type 4 hypersensitivity are mediated by T cells. So it's always going to be T cells. So insect venom, sure, different types of proteins. This is why we're going to be talking about the PPD test, okay? So this is kind of where we're going to get into the PPD test because it's going to be the protein that's initiating this type 4 hypersensitivity. And then, like I talked about, metal ions like nickel, and again, a lot of the times it's added to jewelries. And then like whatever this thing is, you don't have to know this, but poison ivy, literally like poison ivy causes these type 4 hypersensitivities, okay? So type 4, again, it's T-cell mediated. Think CD8 T cells, think CD4 T cells. Doesn't matter if a CD4 T cell is involved, it's type 4. Doesn't matter if a CD8 T cell is involved, it's a type 4. Okay? Doesn't matter if it's TH1, TH2, TH17, doesn't matter, it's type 4. Anytime a T cell is involved, it's a type 4. Okay? But it's delayed because you need to make the T cells. You're not actually making T cells, but you're activating, like you're going through that whole process, that painstakingly long process that we talked about on Friday, where you need to kind of activate go through like the whole selection process of T cells, making them, blah, blah, blah. But as soon as you have a T cell being involved, it's a type 4 automatically. And a lot of the times it's not like weeks after exposure. It's not like serum sickness in that way, but it's just like, it's still something to be careful about when you're reading prompts. Like don't jump for a type 4 if it's like not immediate because of serum sickness. Serum sickness can happen a few days later. Well, usually it's like a week later, but still it can happen a few days later. Type 4s usually happen 2 to 3 days after exposure. If you've ever had poison ivy or one of those, like you would know that you didn't get the rash until at least a day or two later. Usually it takes 2 to 3 days. And this is why. So this is a good example of showing like, all right, this is like an allergen. Let's call this like the poison ivy oil, right? So let's call this. So it's going to be on the skin. So one of the dendritic cells that are present in the skin are going to find it. It's going to pick it up. It's going to travel through the afferent lymphatic vessels, right, to a lymph node, to a secondary lymphoid tissue. And then from here, it's going to present to a T cell. And then from here, the T cell gets activated. It's like, all right, I'm going to leave. I'm going to go find it. It's going to travel through the blood. And then it's going to go back to the same spot that it basically this dendritic cell just came from. So it came back to the spot. And then from here, now we have activated T cells. So in this example, I kind of have like, all right, we're having Th1 T cells being activated. They're activating macrophages. You're getting like just this big inflammatory response happening because the T cells got activated and they're trying to get rid of whatever antigen that they thought was on the surface. So you can kind of think of it as like, again, sensitization and an effector phase. That's why it takes like a few days for it to happen, but that's what's causing the delay. Okay. And don't worry about the cytokines, like that's fine. But even if you do worry about this, they're inflammatory cytokines, right? IL-1, TNF alpha, these are pro-inflammatory cytokines. We're making inflammation. I mean, Th1 cells are literally like a very inflammatory type of cell because you're trying to activate you're trying to activate macrophages. So you're going to get like this inflammation. That's what's causing this skin rash and all of these symptoms of these type 4 hypersensitivities. Okay. Similarly, so here's like another example of it. So this would be like something like, so contact sensitizing agent. So think about like now like nickel within like a like jewelry or a watch. If you wear a watch that might have nickel in it. So what's going to happen is the agent, so again, think nickel, is acting as a hapten. So here is the nickel particle, like this red. And when it enters, so like it's going to just kind of like seep into the skin, right? So it's going to like seep into the skin and it's going to associate with self-antigens. And again, this doesn't happen in everyone. This is why allergy, like no one knows why this happened in this person and not in this person, but it's going to bind to self-antigens. So this is why it's acting as a hapten. It's binding to like a carrier protein to make it really big so it can be immunogenic. And now the dendritic cell is going to recognize it. Now it's big enough to be recognized. Dendritic cell is an antigen, which a Langerhans cell is just the dendritic cell in the skin. It's the specific type for the dendritic cell found in the skin. It's still a dendritic cell. So it's an antigen presenting cell. It's going to present this nickel antigen on its surface via MHC, so via an MHC. And then it's going to find a T helper cell. It's going to activate it to Th1. This is skipping a few steps, but whatever. But regardless, at the end of the day, you're getting tons of inflammation. Again, you're getting IL-1 produced, you're getting TNF alpha produced, you're getting, this is a Th1 T cell, so you're going to get interferon gamma, which is going to activate the macrophages. So you're basically getting the same idea here. It's just in this instance, it's like an actual allergen in a way, like it's that oil that's being picked up by the dendritic cell, versus here it can also act as a hapten. That's what happened. That's just tends to what happened. That tends to be what happens with like nickels and other molecules that are in jewelry and watches. So either way, same response: T cell activation, macrophage activation, tons of inflammation, and that's why you get this. They're trying to show this here. You get like a bump in the skin. Like this is now the skin. It's gonna get red. It's gonna like look like a rash. That's what's causing the rash. Is this inflammation from the T cells? So this is what poison ivy rash kind of looks like. So it looks bumpy, it looks red, it looks really like uncomfortable, and it's this. It's basically this. Just here's the skin cell layer, and here's all the inflammation happening right under the surface because the T cells got the macrophages all annoyed. And this is what happens. Okay, so then PPD, this is like the last part of your type 4 hypersensitivity. So PPD stands for purified protein derivative. Why did I highlight protein? Protein makes it automatically recognizable by T cells. There you go, type 4 hypersensitivity. So part 1, so PPD, this is what you have to do any type of schooling, right? Like they make you do a PPD to see if you have TB. But it's really not like if you have TB, it's like if you've ever had previous exposure to TB. That's what a PPD is for. So you're basically looking to see if you've ever in your entire life experienced TB antigen before. So what they do is they take TB, so Mycobacterium tuberculosis, they take it and they purify certain proteins from it, and that's what they're injecting into. They're just taking purified proteins from TB and injecting that into your skin. So anyone that's gotten a PPD knows that you have to go once for the shot and then you have to go back to get it read. That's why it's a type 4 hypersensitivity. It takes about 2 to 3 days for there to have actually been a response. So again, part 1, you're injecting it into the skin. Part 2, you're checking back 2 to 3 days later, hours. Oh my God, guys, days later. Oh my goodness. Oh wait, no. Oh my God. You can tell I've had a long day, right? This is 2 to 3 days. We're just gonna laugh. We're gonna awkward laugh. All right, 48 to 72 hours is 2 to 3 days. Wow, Christina. Okay. Yeah. So you're checking 2 to 3 days later. Again, it's delayed. So that's what's happening. That's what's going to be happening there. So, and then also, if you've never been exposed to TB, when you go and get it read, it looks like nothing. And you're like, wow, there's a waste of time. I could have told you there was nothing there, right? But what they're looking for is an induration. They're looking for something like this that's like an induration, meaning like a little bump under the skin. They're looking for a little bump because that means this is happening. And if this is happening, then your body is reacting to the PPD, that the purified protein that was injected into you. So here's kind of, again, so here's no prior TB exposure. So here's your PPD, you get it injected. What do they do if you've had it? Great question. I'll tell you, my friend actually my friend had a positive PPD. But so here's what happened. So if you have no prior TB exposure, the macrophage, so it's basically this. If you've had no prior TB exposure, well, actually, I don't have a picture of it, but if you've never had it, macrophages are going to find it. They're going to present it to the T cells. The T cells are like, never seen this before. Like, I'm not going to really waste my time with it. And the reason why you're not getting true T cell activation here is because you're injecting like such a small amount. That's why you're injecting it at this little area under the skin. We're just trying to see if the cells in that little area have ever seen this before. But if you've never seen it before, this is way too small to go through that whole process of memory and that whole process of T cell activation and memory and whatever. So again, that's kind of the idea. Never seen this before. They're like, nah, it's too small. Like, yeah, it's a protein. I can see it, but it's too small. It's fine. So it's like basically the cell's like, nah, I'm not going to waste my time with it. So they don't even generate a memory response against it. Again, I think it's like 0.1 ml. It's such a small amount of liquid. And again, that's the whole point. You're just trying to inject this much to the skin, like just to see like, hey, are T cells in the area? Have you guys ever seen this before? So 2 days later, oh my God, look, I did days here, guys. Hours. Oh my goodness. So 48 to 72 hours later, it should look flat. So you go home and it's going to look just like a normal patch of skin. It's going to look completely flat. But if you've had prior TB exposure, you get this PPD, macrophages find it, they present it to the T cells. It's basically what's happening here in this image here. They present it to the T cells. T cells spaz it out. It's like, oh, I've seen this before. No, we got to get rid of this like ASAP. We have to get rid of this. And that's why it's going to release cytokines. It's this picture is what's happening. And 2 to 3 days later, you're going to get hours. You're going to get an induration at that site. And then they measure it. They use like a little ruler and they measure how big it is, because if it's really big, that means like the T cells are like, whoa, like really, really big response. If it's smaller, they're like, nah. Like sometimes they're like, yeah, don't even worry about it. So what happens if you do have this? Then they do a chest x-ray. So you automatically, if you have a positive PPD, you automatically do a chest x-ray. They want to make sure you don't have TB, like active TB right now. And then if it's negative after that, sometimes they make you do like a quantiferon gold. I don't know if any of you guys have had to do a quantiferon gold. I remember I was like shadowing at a hospital. They did not accept a PPD. They needed it to be a quantiferon gold. And the reason why all quantiferon gold looks at is how much interferon gamma you're producing. So quantiferon gold. I think of the G gamma, but it's like how much interferon gamma is being produced in my body right now. And a lot of the times, if you have active TB, you're going to have granulomas. This is going into a lot of detail, but granulomas, how you maintain granulomas is with interferon gamma. And hopefully that makes sense because you need Th1 cells to activate macrophages to kill TB. And what do Th1 cells release? Interferon gamma. So basically all a granuloma is like tons of Th1 cells, tons of macrophages, and tons of just interferon gamma just all over the place. And it's contained in like a circular little granuloma. That's what a granuloma is basically. So that's why they sometimes make people do quantiferon golds, because they're like, oh, it's more specific for like actual TB. It's not like, oh, have you seen it maybe before in the past? Or are you going to have a cross reactivity with like some sort of antigen you've seen in the past? Yeah, so you've had the TB and so someone's like, oh, I've had the TB vaccine before, so PPD is always positive. So there's a vaccine, it's called BCG. I forget what it stands for. Bacillus I forget, but it's some sort of bacteria that it uses part of mycobacterium in the vaccine. So this is a vaccine. A lot of the times it's people that are like, that live in other countries. They had to get this vaccine before they came here. People in the military, I think, had to get this vaccine. I'm not really sure. But if you had this BCG vaccine, actually, this is going to be, I didn't put this in here because it's more of a second year thing. But if you see this in a question prompt, you're always going to have a positive PPD, always. So if you've had this vaccine, they tell you like whatever, you can never get a PPD because it's always just going to be positive, because clearly you've been exposed to TB antigen because it's literally in this vaccine. Yes, exactly. So you were born abroad. So when you immigrated here, you got the BCG vaccine. And then when you get this, you're always going to have positive PPD. So you have to probably only get QuantiFERON Gold. Next one. This may sound like a stupid question. No such thing as stupid questions. What if the reactive T cells just aren't hanging out in that spot? That's a good question. Basically, it's like there's going to be at least the skin dendritic cells there, because that's where they exist. That's where dendritic cells live. They live just directly, like the Langerhans cells, which are skin dendritic cells. That's where they like love to hang out, and they will usually just always be there. So then they find it, and then they take it, and they go find the T cells, and then yeah. So that's kind of like, it is a good question though. That is a good question. So a lot of the times you'll have these Langerhans cells just hanging out here that are going to pick it up. So you really don't see too many false negatives. You actually see way more false positives with this, because it just say like, I don't know, like your T cells are just like, nah, and they freak out. So that then they'll make you do like a QuantiFERON Gold, or they'll make you do a chest x-ray. Yeah, they also get chest x-rays. Yeah, so so again, you're born outside the US, you come here, you get this BCG vaccine, and you probably get a chest x-ray. And every single time moving forward, you're probably going to have to get QuantiFERON Gold and a chest x-ray every single time, because you can't get a PPD. Granulomas in Crohn's. So granulomas in Crohn's are a little different. So they're actually not involved with Th1. Good call, though. There are granulomas in Crohn's disease, but the difference is this is what's known as a caseating granuloma. And caseating granulomas, again, I'm pretending to be Dr. Sitar with Pathoma, which you guys are going to do next year. But caseating granulomas are only TB. And that's where you get the Th1 cells basically like lining the outside, secreting interferon gamma, macrophages here with the TB inside of it. You're basically just trying to like contain TB. That's a caseating granuloma. Crohn's, and I can't think of anything else with granulomas right now, but Crohn's is a non-caseating granuloma. So non-caseating granulomas is a little bit different of a pathology, which you'll get into second year. Good question. Good catch. Good catch with the granulomas. Okay, and then here's just the last part of the PPD. So here's your PPD. It's injected intradermally, again, where like there should be the Langerhans cells there. So it's going to be within the skin. So they're going to go through like the submucosal tissue. They want to get to like the skin, like where the actual, they want to get to the submucosal tissue. So they don't want to just like get the mucosa. They want to go into like that actual tissue layer. And then 48 to 72 hours later, you measure it, you see how big it is. There is like, again, like I said, there's a lot of false positives. So you can still get a little induration, but that's why you measure it, to make sure it's like, okay, it's within the range. But again, you're getting another induration. So I tried to highlight throughout this everywhere you're going to see like little induration, like see that terminology. So here you would get an induration. This would be a positive PPD. This would mean like you've had previous TB exposure in the past. Induration, you get that with the Arthus reaction. You get indurations with the indurations after the IgE. Sometimes some people get them after the wheel and flare. So just all this terminology. It's just massive. Basically, indurations are just massive inflammation that causes the skin to like go up. Okay. And then we're going to end on transfusion reactions and transplant rejections. Unfortunately, these are a lot of memorization. So I am going to just kind of like read these to you, and I hate doing that. I have like a flow chart to at least help with transfusion reactions. Like I said, I worked in a blood bank lab, so I really needed help with this. But transplant rejections is just a lot of, you need flashcards. You do. You need flashcards. I'll kind of tell you to like what to look out for. But transfusion reactions, I have a flow chart which will help. But again, flashcards are going to be your friend. Are going to be your friend with both of these, okay? All right, so transfusion reactions are what they sound like. Transfusion, you're getting blood. So the first type of transfusion reaction luckily mimics type 1 hypersensitivities. So this is known as your allergic / anaphylactic reaction. And then I put the first aid pictures here just to kind of show you if you need like a visual to associate like I do. You can kind of like think of it like this. So allergic anaphylactic is type 1 hypersensitivity, and it's mediated by IgE, and it's immediate. So it's really great because it's kind of like everything we just talked about with the type 1 hypersensitivity, the IgE, it's all right here, which is great. But what I will say is there's a trick, and I'll tell you at the end. But it's mediated by IgE. Again, it's an allergic reaction. You're basically getting an allergic reaction to the blood. But with this, the patient already has antibodies made. So if it's happening immediately within like 2 to 3 minutes of getting this blood transfusion, clearly the patient already had antibodies. They've already been sensitized. So they're going to be making, they have not always have they been sensitized, because if you're type A, you automatically have type B, right? So that would be an example with, of an allergic. Oh no, it wouldn't. Stop. No, stop. Nope. Cross it out. I'm getting ahead of myself. I'm getting too excited. This does require sensitization. So this is like your classic type 1 hypersensitivity. You need a sensitization event. The patient needs to have made antibodies in the past, and now they're going to be actively, like those antibodies are what's mediating this reaction. Specifically, it's IgE. So again, everything we talked about with the type 1. So again, here's your IgE on the surface of the mast cell. You get mast cell degranulation. So why do I have IgA here? This is one of the most commonly asked questions of transfusion reactions is if you have IgA deficiency. And we're going to talk about selective IgA deficiency. I think tomorrow is immunodeficiencies. And selective IgA deficiency is just someone that doesn't make IgA. So they don't make IgA. You wouldn't think, it's not as big as an issue as you would think it is. It's like the patient might not even know that they have it, besides having like random Giardia infections, right? But they might not even know that they have it. So the number one instance is they get a blood transfusion. Blood has IgA in it. Like it does. You're getting full blood. Like it's going to have IgA in it. So they get blood, and then immediately they undergo fever, wheezing, they're itching. It's like a symptom of an allergic reaction. And you're like, what's going on? That's like your classic patient is receiving blood that has an IgA deficiency. So in the first aid picture, what they're showing is like, here's donor plasma proteins, which is going to include IgA. IgA is in the plasma. And when you get blood transfusions, you're getting blood. You're getting both blood and plasma. So you're getting red cells and plasma. So what happens is you have so you were sensitized in the past, you made anti-IgA antibodies, and now once you get this blood, it can cross-link, and then you get mast cell degranulation. So it's going to be symptoms very similar, again, to allergies, with the exception of you're also going to get like a fever. Does that make sense? So this is, again, so this is your IgA deficiency patient. This is like your IgE-mediated type 1 hypersensitivity, happens immediately. So when you transfuse someone, it's like within like 2 minutes this is happening. Okay. This is what I was thinking about with the ABO. So acute hemolytic. So acute hemolytic, it's like happening relatively quickly. Hemolytic, you're bursting open your red cells. So again, kind of think this is a type 2 hypersensitivity reaction. It's antibody mediated with IgG. That's like another thing with these type 2s. And again, since it's acute, it usually happens either during the transfusion. So kind of similar to this, like it's happening like maybe as you're transfusing this patient, or within 24 hours. So you transfuse them, maybe they're fine, but maybe like 12 hours later, now they're having like these symptoms of, usually it's blood and urine, low back pain. Sometimes they get a fever with it. But with this, there's no sensitization event. The patient already has antibodies made. So again, you have like type A or B blood, like let's see. So in this instance, the host, so the person getting the transfusion is type O. So if he's type O, he's going to have anti-A and anti-B in his serum. And then let's say the donor red cells are type A or type B. So you just made a mistake. You accidentally gave a type O person type A or B. So that's acute hemolytic. These antibodies that the person getting the transfusion, the host, is going to attack the donor red blood cells. Okay? So host antibodies attacked host red blood cells within 24 hours of your transfusion or during the transfusion. Again, it's usually due to like a clerical error, they would say. And then another classic symptom of it is since it's a hemolytic reaction happening, your kidneys are what's kind of filtering that blood, right? So it's going to be filtering the blood that's bursting open anyway. So you're going to have blood in the urine. You're going to have low back pain because the kidneys are like being like injured by this and whatnot. Okay, so acute hemolytic, type 2, antibody mediated. Think type O person gets type A accidentally. And those preformed antibodies attack. Okay. Then you get kind of like would there also be jaundice? Good question. Usually not with acute hemolytic. There's just not enough time. So jaundice is like bilirubin builds up. So usually this is too acute. One of the types of transplant rejections is like, I put my little buzzword B because usually you see jaundice with that type of transplant rejection. It's a good question. Yeah, it's just not enough time for the bilirubin to deposit in like the eyes and whatnot and the skin. Febrile non-hemolytic is exactly what it sounds like. It's you get a fever, but it's not hemolytic. You're not bursting anything open. This is like a weird one. So basically when you store blood, and this is something that another one of my things that I had to do in the blood bank lab, but when you store blood, you're just storing it in like a fridge, basically. That's like what, until they're like, oh, I need 3 units of own egg. And you're like, okay, and here you go. So what happens is it's just sitting there waiting to go be transfused. The blood, again, it's whole blood. It has white cells, red cells, platelets. It has everything in it. Plasma, it has everything. So the white cells that are just sitting in there are like, they just, for whatever reason, start releasing cytokines. And again, it's like during, like, as they're just kind of it's just like a storage thing. Like, they're just kind of there and they're like, all right, let me just start releasing cytokines. It's kind of odd. But anyway, so here's like the first aid image. Here's your donor white cell. So, like, this is a neutrophil, right? Count the lobes, right? Now you guys all know this. It's a granulocyte. So here are your granules. And then it's just going to start releasing cytokines within the blood bag. So within the blood bag, so you transfuse it into the patient. And then it's like, it's just kind of weird. It happens within 1 to 6 hours. Patient has a fever for 1 to 6 hours, but they're fine. There's no hemolysis. There's no like low back pain. There's none of that. This is why in the hospital we would take temperature and blood pressure every 15 to 20 minutes. Yes, exactly. You want to make sure that they're not developing a fever during this process. Exactly. Full circle moment. Don't you love when that happens? You like know what's going on in the hospital. You're like, wow. But what they kind of do to prevent this is they irradiate blood. So they literally take blood and they expose it to UV radiation and they irradiate it. So a lot of the times you'll see if you look at a blood bag, it has like irradiated whole blood. That's what they're trying to do. They're trying to prevent a febrile they're trying to get rid of these cytokines basically. So this would be like if they received non-irradiated blood. That's when you would see a febrile non-hemolytic. So this one, the long name. So transfusion-related acute lung injury, aka TRALI. So TRALI is exactly what it sounds like. It's you get a transfusion, and then basically you get an acute lung injury from it. And what happens is, I always need the image to help me with this. So the donor, so the person donating the blood, has antibodies against the host's white cells. So it's kind of like the recipient or the host. So I always call them the recipient. I think it's easier to visualize. So the person receiving the blood, the recipient, has neutrophils, like just in the body, right? So they have in the body, so including in the lungs. And for whatever reason, the blood, like from the donor, has anti-white blood cell antibodies in it, and they can basically activate these neutrophils. So when you get neutrophil activation in the blood, think about like you're going to see inflammation basically in the lung. So how that's going to present is edema, so pulmonary edema. Patient's going to be coughing. And again, it's minutes to 6 hours. Again, kind of annoying. It's a really long time after receiving blood, but look for cough and pulmonary edema, not wheezing. Wheezing is your type 1. That's like a classic. Did I say it's classic? It's a classic. It's like a little grabber because wheezing is more so associated with like allergies, asthma, right? It's more of like an allergic type thing. Whereas like pulmonary edema and like coughing, that's more of like a lung, like the lungs itself have inflammation. So don't get got with wheezing, okay? And then the last one, delayed transfusion reaction, exactly what it sounds like. It's delayed. So recipient is going to receive blood before. So, oh yeah. Yeah, I was like, what am I saying? So this is in a patient that has already received blood once before, or just like maybe it even says in the vignette like they've undergone regular blood transfusion. So they've received blood in the past. So boom, that's your keyword for sensitization event, right? So they had a sensitization event probably. So they made antibodies against a particular antigen on the donor red cell. So this is not A, B, D. It's not one of those antigens. There's other like really low yield, not important antigens that exist on blood cells. There's like a lot of them. Like an example of one is like Duffy or like Kid or Lewis. Like they're really weird names. You don't have to know them, but they're not A, B. They're not A and B. So there's these weird antigens on the surface of all blood cells. And this is actually what you're testing for with a minor cross-match. So a major cross-match, you're testing for A and B. Minor, you're testing all these other weird little antigens. Regardless, so like, let's say you didn't do the minor cross-match. You were busy that day, so you didn't do it. You give this donor red cell with this foreign antigen to the recipient. They have the sensitization event. And then the next time the donor receives blood with this particular antigen on the surface, they're going to have this transfusion reaction. And how it looks, though, it's not as like intense because these are just minor antigens. It's really not going to be like the massive like blood in the urine, kidney pain. It's not going to be like that. It's just going to be like you get anemia like 2 weeks after the transfusion. It's like less medically like dangerous, basically, the delayed reaction. But it's 1 to 2 weeks after. It's that delayed aspect. So it's 1 to 2 weeks after the onset of the transfusion. You get like an anemia. And you're like, oh, that's weird. Yeah. Here's my little chart for you. So this is actually literally, I copied the one that I used with me in blood bank. In the blood bank lab, I had this tacked to my desk. So did the transfusion happen that day? No. Delayed. It's that easy. It really is that easy. It will never be on the same day. If it did, now I have to go through all the other ones we just talked about. Do they have pulmonary edema and cough? Not wheezing. Pulmonary edema and cough. If they do, that's your trolley. So this is your donor antibodies against host or recipient neutrophils in the lungs. If they don't have pulmonary edema and cough, is there blood in the urine or is there like low back pain, aka flank pain? Yes, that's your acute hemolytic. This is your type 2. This is your antibody against a surface antigen, lysing open the cells, like lysing open the red cells. This is like your accidental, you gave an O person type A blood. If not, do they have fever and chills? That's the best way to do febrile. I mean, you can also see a fever with this, but you're going to see it in addition to other symptoms. Really, with the febrile non-hemolytic, the only symptom is fever. And that's why, like, when you said, like, oh, that's why we tested for fever every 15 minutes during a blood that's literally it, because that's sometimes the only symptom of the febrile non-hemolytic transfusion reaction. And if not, you can just kind of rule of thumb it and be like, all right, it's allergic. But remember, with allergic, you're going to get, like, urticaria, so like itching, you're gonna get wheezing, you're gonna get like symptoms of like it looks like an allergic reaction, basically. All right, good. That's transfusion reactions. Just use this chart. It's kind of foolproof. Transplants, there's only 4 transplant rejections that you should know. These are less, I think, intuitive. These are like, they're gonna take a little bit longer of time for you to completely grasp, I think. But first I want to talk about types of grafts. So there's 4 types of grafts. One is what's known as an autograft. So this is when you take a portion of your own body and you graft it into another part of your body. So yeah, that's an autograft. So they're saying trunk to arm. Okay. But again, it's auto, it's self. So you're taking a portion of your own body and adding it to your own body. An isograft is if you have an identical twin. You can take like a portion of like, you can take like a like an organ from them and graft it into yourself. And then you're like, well, technically isn't that auto because it's like the same DNA? yes, but like you can't say it's auto because it's not coming from yourself. So they're like, let's call it ISO. So that's what isograft is. It's like a genetically identical twin, or like they say, an inbred strain. So if you inbreed, inbreeding, you're keeping the same DNA, right? So that's your isograft. And monozygotic twins are not fraternal twins. So this does not count with fraternal twins. So I have a twin brother. If he donates an organ to me, it's not an isograft. If you have an identical twin sister or an identical twin brother, that's your isograft, and then they donate an organ to you. Allograft is like your typical graft, like that's your typical like transplant. So it's like you do the registry for like I need a lung, you're gonna get an allograft. So this is like requires a lot of like HLA. Remember I said like primarily they're doing HLA typing. They want to make sure that you have at least a decent amount of MHCs that are gonna like line up the same as you, so the least chance of your body rejecting it. But again, allograft is going to be the classic transplant. So from, like it's saying, members of the same species, so Mr. Smith to Mr. Jones, that's your allograft. And then lastly, a xenograft. So if you guys know anyone that had like aortic stenosis or something like that, and they got a pig valve, because a lot of the times they'll transplant a pig valve, like a pig aorta into a human aorta for aortic stenosis, that's a xenograft. So a xenograft is a different species. So it's not like, okay, it's a monkey. It's like anytime you're using like an organ from, I know there's another type of graft that it's like the pig graft, but the pig valve, but it's something like that. Like those are all xenografts because they come from different species entirely. They're not coming from humans. Okay, so this is like easy points, right? So if you were to ask anything on, just kind of remember this terminology. If they were to ask about this, this is like your easy points questions. These, unfortunately, are, again, memorization. I promise you they will make more sense second year. Dr. Sitar goes into this really nicely. He explains the histology behind it really, really nicely. But for now, just memorize it. Just do it for me. Just memorize it. And what I tried to highlight and like underline, I actually only tried to put on here things that they might ask about. So the type of hypersensitivity it is absolutely, you need to know that. Like you need to know the type of hypersensitivity it is. Kind of you need to know what's going on. Like is it the donor antigen, recipient antibody? Like what's going on exactly? This is more so for you, like how long does the rejection take? So like how long does the body take to reject the organ? And then this is more of your histo. This is more of what's going to make sense with Dr. Sitar. I'm not going into it now. I could explain it to you and be Dr. Sitar from Pathoma, but I'm not going to do that because he's going to do it next year and you're going to be like, okay. And then kind of like how do you deal with the rejection? So there's 4 of them. So this is the first one. This is called hyperacute. So hyperacute, this is your type 2 hypersensitivity. So automatically type 2 hypersensitivity, think antibody against a stationary antigen, right? Luckily, this is hyperacute, so it's super acute, right? So it's happening very quickly. It's happening within minutes. So that's another good way. Like you put the lung in the patient and like within minutes, you're starting to notice that they're rejecting it. That's like your hyperacute. And what's going on is the recipient antibodies. So the recipient, the person getting the organ, has antibodies, and they, for some reason, already have these preformed antibodies against antigens on the donated organ. This is supposedly a liver. So there's antigens on here, and for whatever reason, this patient already has preformed antibodies against these. That's why it's happening hyperacutely. But notice it's still antibodies. It's a type 2 hypersensitivity. So again, we're always going to be dealing with antibodies with type 1, 2, and 3. And then how it looks is it's what's known as a white rejection because it's like the graft dies basically. Just again, think about antibodies binding to an antigen, and it undergoes a form of necrosis called fibrinoid necrosis. It means nothing first year, I know, but this is what it looks like. So see this white outline of this, like this whole graft here? This is fibrinoid necrosis, and it just looks like a white outline. That's why they call it a white rejection, because you get like this white line, and basically that's just necrosis. And again, Dr. Sitar next year will go into it. You'll be like, wow, I'm a histologist. But for now, just these are like the buzzwords to keep an eye out for. And you have to take the graft out. There's no saving the graft. You have to take it out. So it's unfortunate. You put the graft in, it's like going well for a few seconds, and then within a few minutes, you're like, I gotta take it out. All right, hyperacute, acute. So it's not as hyperacute. So it's acute. So acute transplant rejection are type 4 hypersensitivities. What does this tell you? T cells. Some way, shape, or form, T cells are involved, right? So that's always what your type 4s are going to tell you. And it's type 4, it's delayed, it's going to happen a little bit later. So this is weeks to months after the transplant. So 2 things that type 4s tell you, T cells, and then how long it's going to take for the rejection to happen. So what happens here is the recipient T cells, so the person receiving the transplant has T cells. So actually, here's T cells within the recipient. And the donor, so the donor, here's like a liver again. Remember, it's going to have MHC 1 and 2 all over its surface, various types. That's why we do HLA typing, to kind of try to avoid this as best as we can. It's going to have this, and it's going to go in, and it's probably going to be fine for a while, but then over time, the body's going to start rejecting it. It's going to be like, nope, the T cells don't like it. And they're like, nope, we don't like this. And so basically, it's like you're making T cells against that donor MHC. And how it looks on a histology slide is you see what's a lymphocytic infiltrate. Lymphocytes are T cells, so it makes sense that you would see, like, again, that's what T cells are, like these little purple cells here. All of these are all T cells up in here. And you shouldn't see this many T cells, like lymphocytes in an area. So this is your lymphocytic infiltrate. It just tells you that T cells are there, right? And then you can actually reverse this if you give the patient immunosuppressants. So a lot of the times that's why they give people that are getting transplants immunosuppressants. So they'll give them like a steroid or like a literal like chemo drug to try and stop this from happening. They try to stop the T cells from getting activated, at least for like a few months. So you can reverse this process by just stopping the T cells from being active. Okay, so hyperacute, type 2, acute, type 4. So chronic. So chronic is both type 2 and type 4. Isn't that great? But, and this is straight up from first aid, it's mostly humoral immunity, meaning mostly antibodies are going to be involved. So that tells me, okay, it's mostly type 2. So chronic, this takes months to years. It kind of tells you in the name. This actually happens to like every organ transplant. I'm not going to lie. Like usually the body's going to eventually reject it. It's not your own self. It doesn't matter if you have perfect MHC lining up. It's like, nah, it's not myself. So it's still going to eventually reject it. But it's going to happen after months to years of having this organ. And mainly chronic, and there's another type that's 2 and 4, but mainly 4. This one's type 2 and 4, but mainly 2. It's mostly those antibodies that are like chipping away at this chronically over time. It's like chipping away at the organ over time, and it's causing rejection, very, very, very slow rejection. So what happens here is the recipient has antigen-presenting cells that are like, I don't know what this is, but I think it's foreign, and they start presenting the donor peptides to the recipient's immune system. And Dr. Sitar refers to this as what's known as obliterative necrosis. And again, just think of this as like you're obliterating the organ over time. And it depends on the type of organ. Like these are way more like second year path things that you look for. But like, let's say you get a lung transplant. This is what it looks like in a lung transplant patient. It's called bronchiolitis obliterans. The bronchioles start to like go away over time. If you have a heart transplant, you get accelerated atherosclerosis. A liver transplant, they call it vanishing bile duct syndrome. It's kind of like the bile duct on the liver starts to obliterate. And then kidney is like, again, this is what happens in every organ. Just chronically, you start rejecting the allograft. So you're going to get nephropathy. You're just going to get like kidney failure over time. It happens in every organ transplant. Okay, but that's what's happening. The recipient's antigen presenting cells are presenting the donor peptide. This is really important to understand because the last one, which we'll talk about, but I want to show you the image. So this is what it looks like. This is like the obliterative necrosis. Yeah, Dr. Sitar will tell you about it next year. Graft versus host disease. This is your buzzword B one I put on here. But going back, so again, really quickly, chronic recipient antigen presenting cells present donor peptides to the immune system. Graft versus host disease are the donor T cells, the T cells in the organ. I think this one is so wild. So like we're donating this liver. The liver has T cells, like it has there's immune cells in it when you donate it. So it has T cells in it. And basically, it's like it's rejecting the body that it's in. The liver's like, nah. Like I put here, it's like, nope, this won't do. Like, I like my old body. Like, I want to go back. It's wild. It's literally, so that's why they call it graft versus host disease. The graft is rejecting the host. So it's so wild. But this one's hard to kind of predict what's going on. You kind of need to use symptoms to predict this. That's why I put the buzzword B here. But rejection is going to vary in time. Some people have GVHD. Some people have GVHD like right away. Some people it takes years and years and years. Some people it's a few weeks. So time's going to vary, but it's still type 4. So again, it tells you T cells. So at least it tells you that. It doesn't necessarily have to be delayed in that way. This was named type 4 because of the T cells. So donor T cells reject the body. Wild. And how it's going to present is this is usually what you always see in vignettes, some combination of these 3 symptoms. You see a maculopapular rash, which, oh my God, I'm going to draw a creepy man, sorry. But you see, so here's maculopapular rash. It's just a rash all over the entire body. And it's red. It's red and it's bumpy and it looks like this. Maculopapular rash, whole body. You get diarrhea and you get jaundice. So this is kind of like your jaundice from before, the person that asked about jaundice. Usually jaundice is more of a symptom of graft versus host disease. Oh, questions. So are donor T cells activating the host B cells? Oh yeah, the donor's T cells are doing like they're literally like they're getting activated by everything in the recipient's body and they're attacking it. So they can be activating the host B cells to make antibodies against the host. They're activating they themselves are getting activated, and like cytotoxic T cells are start destroying cells. Th1 cells are activating more B cells to make it's insane what's going on with graft versus host disease. It's literally like just think of like all these other types of transplant rejection is your body rejecting the organ and in their various ways. This is the organs rejecting your body. So it's all kind of like, it's the opposite. It's like the T cells are like, no. So yes, you're getting B cell activation of the host against the host. You're getting more T cell activation, but it's the people that are doing the big players with this are the donor T cells. They're kind of starting this process. How long does this take to present? Good question. It really varies. I tried to find like a better answer for this, but even first aid says it varies in time. When I was doing practice questions last year too and everything, like you can see a week. So again, with this type 4, don't use this so much for the like how long it takes. Use it more so to remind yourself that the T cells are calling the shots in this one. That's the whole point of the type 4. But it's going to vary in time. You can see a year, you can see 2 years, you can see 10 years. You can see a few months, a few weeks, etc. But more so look for these symptoms. That's kind of why I even put this here, because again, they're going to give you some, like it's going to be very obvious that it's the whole body is being affected. So these are symptoms of like affecting the whole body, basically. All right, that was everything. Do you guys have any questions about transplants, transfusions, fun stuff? No? We just want to do some questions. All right. Remember that if anyone, it's 8:30, so if anyone has to get off, don't worry about it. You can leave. I will stay on and do questions even if no one is here, and then you can watch the recordings. So don't feel pressured to stay at all, okay? But let's dive in. Yeah, this is a tricky one. I remember why I put this on here last year. I was doing like a practice test, like studying for boards, and this question was on my practice test, and I screenshot it and put it for the students last year to be like, look, they ask about it on boards questions, I promise. Okay, I want to talk about this one now, so sorry if anyone really wants to answer, but you guys are like, I think we need to talk about what's going on here, and then maybe you can try again. So this is saying 23-year-old woman, whatever, she's given a large dose of rabbit anti-thymocyte globulin. So she's a human, and she's getting rabbit antibody in just giant doses. Rabbit antibody specifically against the thymocyte, so against thymus. And then from here, this is going in her. Then from here, 10 days later, she's getting these symptoms, these very systemic symptoms throughout the whole body after receiving antibodies. Huh? Huh? No? Clearly, this is a board's question, but I wanted to show you that they do ask about serum sickness. This is serum sickness. So this is your type 3. This is your type 3 hypersensitivity, which is your immune complexes depositing in the tissues. Okay? This was a tricky one. I think a lot of you guys answered this as a transplant rejection, like a transplant rejection, which is fine. I mean, we literally just talked about it, and these are symptoms you might see with transplant rejections. But the key with this is she was given antibodies that weren't even human antibodies. They're against like another animal. They're like from rabbits. And when 10 days later, so about 1 to 2 weeks after receiving this, she has these very systemic symptoms everywhere. This is kind of how they ask about serum sickness. So I wanted to kind of give you, you reject the question. I love it. Type 4, type 2, what type of rejection? But I kind of wanted to give you an example of how they ask serum sickness in a question. This was tricky. Again, I put this first because I was so excited that they asked about it in my practice test. But does this make sense? It can be like a wide range of symptoms, but it's going to be something systemic. That's what they were trying to get at. Arthralgias. If you see arthralgias, oh my God, type 3. Even if you're like not sure, just be like, you know what? It's probably type 3. These immune complexes are probably depositing. Okay. And then the other things, fever, that's a very systemic thing happening. Lymphadenopathy, again, systemic. You have tons of lymph nodes in your body, right? So some of you guys picked, so the erythema on the hands and feet, that's like another good indication of type 3. The polyclonal T cell activation, we're going to talk about this. When do we talk about this? Tomorrow, no, Thursday maybe. This is what superantigens do. Superantigens. So you know like toxic shock syndrome? Toxic shock syndrome is a superantigen that causes polyclonal T cell activation. This is, if you see this terminology, they're talking about a superantigen. Okay. So they're talking about like toxic shock syndrome. We haven't talked about it yet, but just know that that's what you can even write like next to it. If you see polyclonal T cell activation, this super, like they're like interchangeable. Okay. Okay. That was it. I just got excited. So this is serum sickness. This is how it presents. Wide range of symptoms, but again, look at the timing and look at what the patient's getting. This, they're giving her an antibody, so as they're trying to give you a little bit more. Sometimes it can just be an antigen being injected, and then she still has these symptoms. So they're trying to really get at, like, we're injecting an antibody, and it's like, oh, they're making complexes, and then that's depositing. Okay. There's no answer for this, because again, it was a practice test, but okay. Oh yes, this is the last one, I promise, from the practice test. Okay, a few more seconds because that was 90. You can tell that this is like a boards question because it's like you only need like the last sentence or two to answer the question. This is how every single boards question is. Um, okay. Okay. So with this one, all it's really asking is the physician's like, let's give Rogam. And it's like, why? What is Rogam gonna do? What's it gonna prevent in the mom? And Rogam itself is gonna prevent anti-D antibodies. So remember, Rogam coats the fetuses or the fetus and newborn fetuses or the newborn's red cells. It's going to coat it like this. It's basically coating the anti-D. Sorry, it's coating the D antigen, so that if this gets to mom, she doesn't see it. Right? So she doesn't see it. It's fine. So mom will never make D antibodies. Does that make sense? Rogam. It coats the fetus's red cell D antigen to prevent mom making D antibodies. Cool. Awesome. All right. Moving on to the non-boards questions, I promise. Okay, about 90 seconds coming up. Okay, so with this one, we got penicillin. So almost immediately with penicillin, when you see like a penicillin and then they're asking a hypersensitivity like question, you want to think again, there's two ones you want to think of. You want to think of type 1s and you want to think of type 2s, right? So type 1, type 2s, and type 3s. Oh my gosh, you want to think of everything that's not T cell mediated. So I'm sorry for my T cell people. With the maculopapular rash, remember that this is more so for graft rejections. When you see like maculopapular rash with like fever, with jaundice, with stuff like that, that's more so for like your transplant rejection. That's more T cell. But for penicillin, you want to think of one of the antibody ones. So with this, what kind of gives it away? What gives it away is that it's so she's treated with penicillin G, doing with a penicillin sensitive organism. During her second week on therapy, she develops fever, red maculopapular rash, pretty much all over her body. What does this sound like again? So this is going to be, yes, this is your serum sickness again. I know. I'm so mean. I know. But this is your kind of like your serum sickness again. It's a week. The timing's good. So it's like 2 weeks into the therapy. She was previously sensitized already. And now she has like these systemic symptoms again. So this is going to be like your IgG. This is going to be your going to be your immune complexes. This is going to be your. A lot of you guys were split. So you guys like being like, a lot of you guys were like split between A and D. A lot of you guys were, but I wanted to I liked this specifically. Like with this is so classic, and this is what I wrote here. Classic acute penicillin hypersensitivity is mediated by IgE and histamine. So that's what I'm saying. Like a lot of the time, it's like penicillin. When you have this allergy to penicillin, usually it's IgE and histamine. But the timing was critical for this. 7 to 10 days later, this is like more of like these immune complexes that are depositing, causing these widespread symptoms. So I liked this answer though, because I wanted to bring your point here. Drug reaction occurring days to weeks after exposure, involving just many organs, many things, skin, joints, kidneys, almost always, this is Dr. Ryan saying it, not even me. Dr. Ryan, almost always a type 3 hypersensitivity. And that's what I kind of tried to put on that slide to really try and emphasize it. Like penicillins are classic, your classic type 3 hypersensitivity. This is like your classic serum sickness with the antigen antibody deposition, like the complexes depositing. Her first sensitization was like the first week basically on the therapy. So the woman is treated with penicillin G for endocarditis during her second week on therapy. Like it took her that long of a time to make these antibodies. And now it's kind of like if I had that chart, remember with the serum sickness chart, where it's like we inject the antigen here and the levels skyrocket and then they start to wane. But then at the same time, we start to make antibodies. And then in this middle zone, this is where we get the complexes, and this is kind of what's causing these symptoms. This is like classically what penicillin does, and that's like what causes your serum sickness. So again, look for skin findings, joint findings, kidney, just anything. And it even says protein in the urine. That's a kidney thing, right? Yeah, no problem, no problem. These are tricky. Again, that's why I wanted to show you questions on them to kind of show you like these are kind of the rules of answering these types of questions. And kind of like what we've been talking about a lot, it takes several days for B cells to do this. Like it takes a while, again, with that chart. This is over days, right? So it's going to go up and then it's going to come down and then the antibody's going to come up. This is taking days to happen because it takes B cells a little bit of time to make the IgG antibodies that are going to eventually bind to the antigen and make these, which is penicillin in this case, and then it's going to make the immune complexes. Okay? But again, you're not like a lot of you guys did pick A with IgE and histamine. That's fine. You can think of IgE and histamine with this, but again, look at the timing. This is immediate. This is going to happen like as soon as you take it within like 30 minutes, you're going to get these symptoms of histamine release. And then it's saying like this is resulting in a less dramatic clinical picture. You're not seeing like signs of anaphylactic shock. It's just like more of like these systemic symptoms. Okay, serum sickness. We love serum sickness. Okay, next. Okay, minute and a half. All right. So this one is actually the one that I told you we were going to talk about later. I was trying to not give it away. So a lot of you guys were split, and exactly how I wanted you to be split, because that's what I wanted to do with this question, between B and D. So a lot of you guys are split between this. So he's got an induration. And remember I said it's kind of, and again, it's 6 hours. So remember late type 1 hypersensitivity happens within 6 hours. Type 3 hypersensitivity, specifically your Arthus reaction, happens from 4 to 10 hours after an injection. So how do you kind of know which one this is? Well, this one is going to be an Arthus reaction. So this is going to be an Arthus reaction, and I want to let Dr. Ryan do the talking with this. So he's basically saying with an Arthus reaction, having injected insulin, it's going to be type 3 hypersensitivity. The complexes are forming, they're depositing, and then you're getting deposition blah, blah, blah. Local reaction occurs due to preformed antibodies against the antigens. So this is kind of how you know it's a difference. So this man has formed antibodies against some component of injectable insulin, either the insulin molecules themselves or some sort of additive. So when he first begins his new insulin, that was kind of the key with this. He was switched to a new long-acting formulation of insulin. They told you he just switched insulins now. No reaction occurs because the antibodies have not yet been developed. After several days on the drug, antibodies are present to react, which leads to the Arthus reaction. So this is going to be your Arthus reaction. The time frame is key. So the time frame, the nodule develops 6 hours after injection, too slow for type 1, too fast for type 4. Reaction must be against a component of the insulin. Okay, so again, how do I know that it's type 3 and not the delayed type 1? The delayed type 1 is always, maybe I should put this on the slide. I think I might do this because this happened last year. It's always going to be preceded by you're going to get the wheel and flare first, and then you're going to get the 6 hours later, you're going to get that induration. So it turns from, that's why I tried to put the pictures side by side. You get the wheel and flare after however many minutes, and then you're going to get the big giant induration 6-ish hours later. Does that make sense? So you can even go right on that slide. I think I'm going to add that to this PowerPoint. But it's always going to be preceded by this. So everything's kind of the same. That's why it takes a little bit of time because you need to make the new antibodies, right? But he's not having that early type 1 hypersensitivity reaction. It's almost like he just jumped along to the late, right? And that should be more of an indication of an Arthus reaction, which is kind of what he's saying here. 6 hours after injection, too slow for type 1. Because again, it would need to be the first type 1. It would need to be the early type 1. You can't just jump to the late, if that makes sense. Make sense? So I told you I would have a nice question to kind of show you the difference between the two. But yeah, it's good that you at least picked up, this is going to be an induration. This is your classic. This is either going to, in the indurations are in the late type 1 and the type 3 with the Arthus reaction. But this is your Arthus reaction. Okay. So even if you picked B, that's good. You're at least you're remembering. But okay. There's, I think, 3 more. Yeah. These, you guys can even refer back to your note. These take a little bit of time. I shouldn't have even put these in here. No problem. Yeah, leave whenever you have to leave. No worries. Okay, about a minute and a half. You guys are pretty evenly split again, which is good. So this one is, again, always look at timing for transplant rejections. So 2 hours after getting a kidney transplant, 14-year-old girl has some lower abdominal pain. Okay, tenderness to palpation. She got edema, serum creatinine begins to rise, etc. So what's going on here is, again, the timing's going to be key, but this is your preformed antibodies against the HLA. So it's the one picture that has the recipient antibodies against donor MHC. That's kind of what's happening here. And it's patient is rejecting the donor kidney. When it happens with less than 48 hours after transplantation, this is your hyperacute. So again, the timing is going to be absolute key for this. So hyperacute is going to be this one. hyperacute graft rejection, preformed cytotoxic antibodies against donor HLA molecules. Okay, so antigen antibody binding activates complement. You get all sorts of stuff. This is a very, I think I'm going to take this out because this is, you're not going to have to do, it's going to be a lot more simple. It's going to be a lot more simple for your exam. I just kind of wanted you to see that they do ask about transplant rejections, but the other most commonly asked one or answered one on this was the T cell. So like that would be like your graft versus host disease or whatever. But again, it's that, or sorry, not graft versus, that would be like your acute transplant rejection. And then that would not be 2 hours. That would be like a little bit later. So it's really timing. Timing is going to be really the key with transplant rejections, is kind of the point I was getting at. Okay. Guys, don't get got with this. Make me proud. Okay. Okay. About a minute and a half. So a lot of you guys got. So I'm really happy though. So you won't get it wrong on the exam. A few of you guys got it. Very proud. Very proud. It's because you hear me in your head saying not to get got. Okay, so the key with this one is, what type is this? This is type 1, right? So this is going to be your classic, like you have the early type 1 symptoms and then you get the induration. So you're going to get like the wheal and flare and then you're going to get the induration. The mast cells and histamine are what are activating the wheal and flare. This is what's responsible for the early type 1 symptoms. So this is going to be up here. So minutes after being stung, she developed redness, swelling, and itching. That's because of the mast cells and the histamine. But they want to know what's causing this induration now. So it's like, what's causing this induration? And that's going to be macrophages and cytokines. And I even put it on that slide. I put it on the slide where we talked about it. It's like a little line. But that's another like thing I wanted to talk about is like the wheal and flare. The early is your mast cells histamine. 6-ish hours later, when you get the induration, that's now the macrophages and the cytokines being released. It's on that slide. Just kind of go back to that slide, highlight it, be like, this is for the induration. Add the little note. I think I'm going to update that slide, make it look more fancy before I send this to Sarah. But mast cells and histamine are responsible for early, macrophages and cytokines late. Early always precedes late. But all of you either picked A or B, I think. I'm pretty sure all of you guys either picked A or B, so you're good. You're still thinking within the type 1 hypersensitivities. So good. Again, you just learned this today, so it's fine. But key point, I always like these key points from Dr. Ryan. So indurations can happen in 3 ways. It can be late type 1, which we talked about. It can be arthus, which is your type 3. Finally, it can be a type 4. And again, this is also kind of why I went back and I highlighted every instance you get an induration. So you get late type 1 induration, a type 3 arthus reaction, you get an induration, and then the type 4, delayed hypersensitivity, you get an induration. So again, how do you know which one of the indurations it is? And it's always timing. Always going to be timing. And specifically with type 1 versus type 3, like the late versus arthus, look for if the late type 1 is preceded with an early type 1, which is going to be your classic itching, redness, swelling. Okay, but 6-ish hours or so afterwards, late symptoms develop. That is your induration at the site, and that's going to be, again, just neutrophils coming. It's always the same cause of, like, the induration is always caused by macrophages and neutrophils just getting annoyed and just releasing cytokines and stuff. But again, since we're talking about the type 1, specifically in this example, that's why it's not T cells. It's not this one causing the induration. And it's not this one. Well, it's not this one because this would be the type 3, the Arthus reaction, which is not happening here because we had that early type 1 first. So we really were only, for the other third cause of an induration, it really could have only been this one. But alternatively, when you guys were like, well, I know it's a type 1, so you picked one of the 2 type 1 answers, and that's good. We just learned this today. But does that make sense? I really tried to, clearly I picked a lot of questions where we're talking about this, because these are the tricks. These are the tricks with the hypersensitivities. Okay. Last question. Okay, let's chat. Let's chat. Guys, you guys made it harder for yourself. So, this gave you the answer in the question. Can you believe it? So, this is saying you get the diphtheria. It says toxoid vaccine. We talked about it yesterday. Toxoids are proteins. It tells you that anyway, so it's fine. So, testing shows the voice cells cannot mount an antibody response, but it's because of a mutation in one of his HLA genes. They tell you, what do HLA genes code for, guys? What do these code for? MHCs, yes. So literally, this is like basically saying like it's caused by this. So it's like a failure in which of the following is probably what's causing this. It's going to be an issue with one of these MHCs. So you guys want to try again? Who didn't pick C or D? Do you guys want to try again with this? Because all of these technically could be, like, they really all could be right, but it's this key right here. Like, they're telling you it's an issue with HLA. And remember I said the HLA genes are what code for MHCs, so this is saying there's an issue with one of these MHCs. I know, and when I did this one too, like, if you're reading too fast, you're just like, oh, wait. Okay, now we're getting it. Now we're getting it. Good. Okay, perfect. Okay, so this one is, yes, MHC class 2. Very good. Very good. A lot of, I think, some of you guys keep C. Maybe some of you guys kept C. That's all right. That's all right. You got one of the MHCs the first time, which is fine. So this is basically saying like that, like it's actually, I'll just let you know what Dr. Ryan. So he got a peptide-based vaccine, did not mount an immune response. The protein antigens consumed by antigen presenting cells presented to CD4 positive lymphocytes. But regardless, HLA genes code for proteins of the MHC and MHC2 complexes. Therefore, lack of the response must be due to MHC processing. Okay, so that was kind of just like reminding you guys that MHC and HLA, like that's what HLA is coding for. They're coding for MHCs. So if they've got an HLA gene defect, it's got to be one of the two MHCs. Since this is not a like a live virus, it's not gonna like infect cells on its own. So it's not going to be presented by MHC class 1. Remember, MHC class 1, these are your endogenous antigens. So endogenous antigens are going to be like your viruses. They're things that like actively replicate in the cytoplasm. Remember, and then they get loaded in the endoplasmic ER, rough endoplasmic reticulum. Oh my God. The rough ER, and then they go to the surface, and then CD8 positive T cells. Anything exogenous, toxins, bacteria, fungi, anything that's not actively replicating within cells is going to be presented by an antigen presenting cell with a class 2 MHC. So this is a toxin. It counts as an exogenous antigen. Okay. All right. We're going to end there. I have one more question from that practice test, but we're not going to do it because it's tricky. I'll put it on there though. You guys can do it. Oh, you guys want to do it now? You guys want to do it? All right. Well, just know that it's tricky, but it's a review. Here you go. See, they ask about molecular bio on boards. She had enough. I don't blame her. You guys have actually all answered the same thing, so I'm actually kind of curious. Oh, we got one. Great job. But I'm gonna show you like kind of an easy way. So let's just go over this like together because this is like a weird hard question. So this is basically saying so lane 2. So Southern blot looks at DNA. So all this thing is saying is like here's a lane with DNA. We have one band of DNA in this cell, in this organ, in this organ, in this organ, in this organ. We have the same DNA because this is how you identify DNA. But why in the bone marrow do we have all of these random DNA bands? Like, why do we have different size bands? Why do we have smaller bands, bigger bands, large? Like, why do we have all these random bands in the bone marrow? So if you, even without reading the question right, you can just straight up be like, which of these processes it must be happening in the bone marrow, right? So which of these is happening in the bone marrow? So in the bone marrow, you get I'm going to give away the answer, but yes, good. VDJ rearrangement. That's like pretty much the only process on here that's happening in the bone marrow with B cells. Does it say that this is B cells? Immunoglobulin constant region. So you can kind of assume that this is something to do with the antibodies. So with this answer is gene rearrangement. This is C. So this is VDJ rearrangement. A lot of you guys went for somatic hypermutation. Totally get why, because you were like, okay, here's a band for one, and then I'm going to mutate it a little bit to make it more specific. But the big key with this is you're in the bone marrow. You don't get somatic hypermutation, affinity maturation. You don't get these processes in the bone marrow. This is in the secondary lymphoid organ. Remember, it goes back and forth, follicular helper T cell, dendritic cell, but it goes back and forth, back and forth. That's your somatic hypermutation, affinity maturation. I could have also seen RNA splicing because that is also happening. But again, this is a southern blot, right? Southern blot is DNA. So yeah, good. Yeah, snowdrop. Yeah, so if you guys don't know snowdrop, it's going to be southern blot is for DNA. So like snow, I don't know how much you guys are familiar with these lab techniques, but snowdrop, thank you for putting this in the chat, reminding me about it. It's like a throwback. So southern blot looks at DNA, northern blot looks at RNA. I don't even know what O is nothing, right? Yeah, I don't think that's anything. And then western blot looks at protein. Okay, but this tells you it's DNA. So that's why RNA splicing was not the answer, even though RNA splicing does occur in the bone marrow. Somatic hypermutation happened in the FAB region. Yes, that's another good pickup. This is the constant region, right? Somatic hypermutation, affinity maturation doesn't do anything with the constant region. It actually just only affects the variable region. The VDJ rearrangement, right, happens within the variable region because we're trying to make a very like specific antigen binding site on this antibody. RNA shouldn't be targeted by restricted enzymes. Exactly. Restriction enzymes they recognize DNA, and that's what's shown here. Where is it? Restriction enzymes, they look at DNA, they don't digest RNA. So that's how you could have ruled these out. This one out as well. Somatic hypermutation, variable region, not happening in the bone marrow, out. Affinity maturation is basically like you're picking the somatic hypermutations that worked well. They kind of go hand in hand. Doesn't happen in the bone marrow, doesn't happen in the constant region, out. Apoptosis, I think that this is also like a good kind of get answer because you're like, okay, well, maybe it's like this is a dead cell and then this is a dead cell and then this is, because DNA would still show up even if the cells died. But again, it's kind of getting at like there's no technical errors that are occurring here, but which of these processes are happening in the bone marrow? And remember, when we do VDJ rearrangement, we're splicing out DNA. Like, so we get D and J that happens first. It splices out that intervening DNA. Then we get the V combining with the D and J, splice everything out in the middle. So you're getting different DNA sizes as you go through this process. It's like really interesting that you're losing DNA but becoming more specific. It's such an interesting process, VDJ rearrangement. But this was tricky because it was like a lab. It was like molecular bio, like in combination with it. But does that at least make sense? Like why you see all these are all basically like maybe this is a band that underwent this is a band that hasn't done anything yet, like no VDJ rearrangement. It's like a really large band of just normal DNA. This one maybe did a little bit more, then a little bit more splicing, a little bit more splicing, blah, 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 until you get like all these different sized antibody, like antibodies within the bone marrow. Okay. Molecular bio, right? Yeah. All right, let me stop recording.